there in bringing up to this entire process can be met very easily if it is been planned well in advance. Now we need to understand that in order to implement the innovation, what actually the things are required. The most important at present time, if we see, if you don't have technology, then I don't think that anything is possible, whether it is industry or it is in academia. Without the technology, we are helpless. And that's how we have already seen that the technology usage in teaching, or technology usage in business, or technology usage in life has become very, very important. And this we have already learned a lot of lessons. If you are lacking behind in the adaptation of utilization of the technology, then you stay behind. You stay, you are not within the competition. And this is what I have highlighted in many of my lectures, my presentations, that technology is one thing. It is not just related to the job or to the industry. It is even very, very important to the academia. And this we have learned the lessons that how we have faced challenges. Organizations which were not fit in enough with the technology, not able to impart the quality teaching during the lockdown that we have faced. And probably I this is one of the slides which always I would like to present to the teachers, that the entire world when it was under lockdown, and one of the major in the UNICEF website they have given is that class attendance absent 168 million children. This is one of the the message which was heartening to feel that yes, so many people could not able to attend. Probably we are talking about the 168 million children of being remaining absent in the class is of the physical. But when it comes to digitally, even it was almost the same, because many of the organizations and institutions were not prepared to take up the challenge of the lockdown that has occurred, because technology was not part of the teaching. That's how that has become one of the challenge in order to accept. And probably by the time of the second lockdown, we were prepared up to 70%, I can say, but still 30% of those institutions which could not able to adapt to the technology has affected a lot. But we need to understand why I'm talking about through the innovation and research. When it comes to academia, I do feel that the first innovation that the teachers need to adapt is learn technology, adapt it into the teaching methodology. Then only you are going to attend to the next second part of research, until and unless you don't not adapt it to your innovation in teaching. That's how we need to adapt, and we need to take up. And as a teacher, I've seen that the technology, the way it is adapting and bringing up the innovation in library, if you see, we were very much accustomed to take the teachings, the utilization of the libraries in the physical form. Now entire thing has been converted into the digital form, where ebooks and e-libraries have become very much common part of the the academic activity, research activity. If you are not adapting, you are not accepting to this technology, probably this is quite difficult. You can't. I must say that many of those books which are not available physically in the libraries have been made very much easily accessible because of this e-technology that has come. So e-libraries is one form, e-journals, e-textbooks, all these have become very much easily accessible through various digital platforms. So this is one form of innovation that has happened in the teaching section. And if you see, even in the classrooms, which was we used to adapt in the olden days, where the teaching used to go on a blackboard or on a whiteboard marker, used utilization. That was the limitation of the teaching. The far understanding of the people. But this lockdown has made the people to understand that teaching is not just about the blackboard, even it is beyond to it, where a student can sit at home and teacher can be at any point, the region, and can reach out to mass. One such example is that this summit 2022, Pharma Summit, where the attendees, mass all have attended. They are all from the different regions and attending to it. And I myself from the Bangalore attending, delivering my talk to the mass people from all over the world attending to it. This is one such example. That's how. But the institutions needs to understand now whatever the classroom they are creating, that should also have not only just we used to talk that college should have the classroom, the college should have the blackboards. Now the later we started talking that the college should have the should be a smart campus. That means they need to provide the internet connectivity, accessibility to the students so that they can refer to some of the motivational videos, to some of the research collaborative videos, such type of activities. But now the institution should also provide not just the smart classroom, even the digital classrooms or e-resource classrooms where the teacher can enter with a multiple screen classroom where they can reach out individually to the student on the screen and deliver the talk. That's how the digitalization. I do see this the the studio classrooms which are now become very much common. So each one of the institutions which are imparting this education should look into it and should bring up this adaptation. We have seen that Western countries have long time back adapted, and that's how for them it was not a challenge. But Indian system needs to think and should adapt to this. And now, when it comes to teaching, when it comes to pharma system, I must say that the digitalization, even in businesses, have started. So this digitalization is nothing but part of the innovation which it has been used. Like if you see the pharma means the community pharmacy, the hospital, or the clinical, or the teaching, or the industry. But in all these sectors, if you see it now, in one such sector, I have shown that the teaching has been changed into the digital form. Now, when it comes to business part, where you can see that there is an e-verification system adapted by the hospitals where they get to know the type of the medicine they are getting the medicine, dispensing it to it. So that's how the e-verification system. And these are all certain things which I am talking is of the digitalization of the business. So now there was a time when the doctor used to write a prescription, patients used to carry that prescription to the pharmacy, and the pharmacist used to honor that prescription. But the time has gone. Now the patient sitting at home will visit to a doctor digitally, that is through telemedicine services, and where the doctors identify the problem and then he writes digital prescription, that is e-prescription. That's how that e-prescription comes to the pharmacy store. And this pharmacy, through that e-prescription, after verification, dispenses the medication through again telepharmacies interacting with the patient and counseling to it. And then finally, I must say that the medicine which is now probably being taken by the third party to deliver those medicines to the patient house, but need to adapt more of those technologies probably in future. I see that probably the adaptation of drones in providing the services of medicines, as we have already seen, it has come in delivery of the foods, but medicines has not yet started. Probably is one of the area where the innovation is required, adaptation is required. That's how. And we have already seen this technology has taken over in everyone's mind, and we have become more cautious of health because of the more of the lethargicity of living of life. I can say that like one such is the digital apps. There is every layman, or I must say layman because those who are not aware of medical terminology, those who are not aware of whether medicines or related to diseases are now getting educated through various technological things that are available, including digital apps. The person, when he walks, counts that how many steps he has walked. That person uses the technology or the digital apps to check his pulse. We have seen this during this lockdown period, checking of the oxygen levels, checking of the blood glucose levels. Yeah, accuracy is one of the question mark, but still the innovation of adaptation and bringing up that into the practice is one of the challenge, and that is developing. So that's how this change that is happening should be accepted and should be used it in order to bring up more proper services and safe services for the patient. And I have spoken in many of the platforms that wherein 3D printing or the AR or VR, that is augmented reality or virtual reality, or even artificial intelligence, these are all certain things which are still more going to come. We have never heard of these technical terms when it comes to the teaching or coaching to our pharmacy students, because we used to imagine that these are all certain related to the engineering sector which we may not need, require to study. But I must say that these are all going to be part of our life system. Not just the pharmacy, maybe every medical fraternity needs to understand these technological terminologies. We have to understand and then we have to adapt. That's how it changes. And this innovation is very much is part of the profession now. We need to adapt and we need to take further to it. Okay. And 3D printing. These are all what I wanted to tell. Now, pillars of innovation we need to understand. When we want to adapt it, there are 8 pillars, which is very much important in order to adapt the innovation either in institution or into the organization. One is the process. Like we need to understand that the innovation comes with the ideas, designing, implementation, and adjusting innovation strategy, governance, performance management, measurement, innovation technology, and team structure. That's how when it processes through first with the designing implementation, then only you reach out. And people here finding the right talent and training is very important. That means when you are having the idea, when you are having the people, then you execute the next level of having a proper level of project which you plan. Maybe short sprints or starting small or growing when needed. So timeline, how you need and at what time. These are all certain things which you need to understand and attend to the problems. See, when any research comes, we need to know more about the problems than the benefits, because we can able to.
Sorry, Salahuddin, sorry to disturb you. Your
Yeah.
slides are not moving. So
Oh, I'm sorry. Wait.
now it's
I don't know. You should have told me. No?
yeah, you were in the first slide
Is
only.
it fine?
No, you are still in first slide.
Please, little bit. Can you? I'm not able to hear you.
Yeah, now we can see. Yeah.
No, no. I have to take it out. Okay.
You are in a second slide right now?
No, I'm not in second. Now you tell me. It's
No,
moving.
no, it is still not moved.
Just wait. Just wait. Now
Yeah. You are in a third slide, isn't it?
it is moving.
Yes, yes.
Okay. Uh, sorry. These are all technological things. We are talking about technology. Okay. So I have covered this innovation implementation. I have covered about what the world. Okay. And innovation in teaching, innovation in businesses, to which I was covering to it. And I'm talking about the pillars of innovation. Hope my slides are visible. My slides are visible.
Yeah, your slide is visible. Third slide is visible. You go to
Sorry.
slideshow, Salahuddin. Go to
I'm putting it. I don't know. It is in slideshow mode now, whether it is visible.
It is visible, but not in slideshow.
Now,
It is not, but go ahead. It's okay.
okay,
Yeah,
no problem. There must be some technical
technical
issue. Okay,
issues. Yeah.
I'll try to take up so that Um, I'll just go across to the slides. Uh, Hope my ninth slide is visible. Okay, so probably what I can do is that every time I need to. Okay, my ninth slide is visible. This pillars
Yes.
of innovation.
Yeah. Yes. Yes.
Yeah. Okay, so I was talking about the process, people, projects, and then problems. Before going to any of the project, you need to understand the problems and solutions to it, which is very important. And that's how the true innovation solves the real life problems. So when you are planning for innovation, you need to have these, the pillars in the life. Then look into the priorities when you are planning for a long term or short term. You need to have the priority of it and progress of the project when you take up, whether it is short duration or long duration, and places where you wanted to implement and to what form, and then partnerships. These are all certain things which you need to adapt and you need to take up. And most important thing, you need to understand that the research is a culture of research provides the supportive context in which research is uniformly expected, discussed, produced, and valued. So when you are planning for any research, so I was talking about the innovation, where you have seen that the pillars that was very important for it. Now, when it comes to research, we need to have a culture of research that provides supportive context in which it should be uniform and it should be discussed and produced and valued. That's how the research comes into it. Sorry, I'm just going back and coming to it because the presentation is not moving. So accordingly, I'm taking it and I'm running out of short of time. I'll try to see that I'll complete it fast. So research, we need to adapt to it, and this is very important slide. Just have a look to it. And most important to understand that the culture of research and the future of the science system, how it is going to be when we are talking about innovation and research. You need to understand the three aspects of the ecosystem of a research is that the person self, when you are planning anything, you yourself become very much important to it, and then the employer and the funder. These are all certain things which we need to understand when we take up the research. The employer also becomes very much important, and even the funder. We are talking more of. In fact, yesterday I have met one of the the funding agency. Now we are talking about not only the innovation research, okay, startups, entrepreneurship. We are also talking about the angel funding or venture investment. That's how the culture has changed. When you are planning anything to go up to the new research, you need to see that even there are people who are ready to invest money into your any ideas that come. That's how the implementation of such a system becomes very much important. So we need to understand that when you take up the research, these are all certain things where you can access to the system through open access. You can adapt to the research by currently adapt going across the system. Probably this entire picture gives the highlight that how the research system needs to be. More important, you need to understand that people with flexible nature takes up goes to the success. That's how you need to understand it. Okay, so understand to it. Now, pillars of research, small steps can make a difference. We need to understand. So I have spoken about pillars of innovation. I'm talking about pillars of research. We need to understand that the small steps can make a difference. So in an organization, when it comes, you need to plan with smaller things. Take those ideas, establishing support systems that can boost the morale and enhance a positive research environment within the institution or organization. As a teacher, I do feel that the head of the institutes should take this initiative where they should keep encouraging and work for the support of the system. Means some of the facilities that are missing should be provided, should be encouraged, and should be known to see that that boosts the available facilities, boosts the morale of the employees or the researchers. ensure everyone is on the same page. You take up, ensure that everyone is on the same page, and research cafes are an excellent example way to share the best practices. That means you create an environment where people go, even if they sit, they start talking about research. That's how many of the organizations have adapted research cafes, where they go for a cup of tea, they have a discussion panel, they have a board where they look into research activities. Organization, department, and team leaders leading by example in promoting an excellent research culture is very important. We have leaders who are they themselves driving the force. We have another speaker, Sunita Reddy. I know that she is one of the the researcher who is promoting the excellent research culture in JNTU. That's how it is increasing a lot of activities over there. So such type of things should come into the institutions. Discuss training gaps for all team members. You need to see that if your team members, as a leader, if your team members, as a researcher, if they are having lack of knowledge in any of the area, you have to see that that training or knowledge updation should happen by providing them the accessibility to it. In embedded research culture at institution level, very important. Many of the undergraduate institutions think that they don't have the facilities, so they cannot promote. Probably this is wrong, because now the Pharmacy Council of India has brought new regulations in undergraduate that promotes the research even at undergraduate level. So it's not just pertaining to the you know the research where people are taking only about the review of literature. Probably come out of it. It's not literature research. It is about the practical research that needs to be adapted. So it has to be brought in. Now, coming up to understand that what is most important is that for the research and innovation, it is the communication, which is very important. So today, if I am able to join on time to this summit to deliver my talk, it is because of the communication. So one thing we need to understand, communication is the main tool that helps the people to achieve to the biggest targets, and that's how the communication from the summit people, okay, Pharma Summit people of the BioLeaks as well as APR has helped a lot all of us to come on one platform. That's how we are doing it. Okay, and we need to understand that when I say about the research innovation, I also say about startups. People should take up these startups. That is all about something innovative ideas. If you have take up into a new business ideas, and I'm always encouraging that people should take up the startups if they have the ideas. What is that idea? Startup is nothing but it's a new business wherein you have something in your mind that you bring it up, merge it up as an innovation, use technology to it, and apart from these things, you need to adapt the passion attached to it, and that's how it leads to the startup. And you have seen many of the successes of the technologies that are happening, and even the government of India is encouraging for startup to digital media to bringing up startup agencies, even through various institution innovation councils. Okay, so many things. Why we have to take up? We have to understand that 35% of the population is young India. That means we have the immense potentiality to grow to the next level of being the pharma leader of the world. That's how we can take up the more. And if I say why young India, because if you see all those startups that have started, probably I'm giving examples of other than pharma, because these are all certain newer ideas they have taken, whether it is Paytm, Flipkart, Snapdeal, Ola, or Oyo. These are all certain the innovations that has been adapted by these young entrepreneurs. And because of that, if you see the business, the market that has turned up is quite immensely acceptable. And why we need to know all these things, it is that all it takes is the innovation that makes you an odd out of all the events in the market. That's how your any idea of having about the research should be planned and it should be executed into the platforms. Now, what is important is to know is that you have to have your own research team. You need to set up, encourage the young members to think big, to achieve bigger, and provide them support and handholding for their growth. Whole chain program and events on startups and entrepreneurship in order to bring up the innovation and research into it. That's how the organizations should plan up and should see that you can do it. And most important, before I conclude, I would like to say that uh, you need to have three things to create a successful startup. Start with good people to make something customers actually want and to spend as little money as possible. That's how you are going to be to bring up your innovation into startup. Now, positivity, I would like to spread over here is that have pride in how far you have come and have faith in how far you can go. That's how you need to plan up. And most important thing is that it is not the strongest or the most intelligent that will survive, but those who can best manage the change. We need to accept it to the change, adapt those innovations into our research to bring up the most important changes. And that's how I always say, dearest past, thanks for all the lessons, and dear future, I'm ready. That's what I wanted to say. When I'm telling the dear future, I'm ready, it is a technology that is going to be the most important. Let's accept it to this all innovation to our research by adapting technology, because a modern tomorrow makes a better nation. That's how I do feel. And for this, I would like to conclude by saying thank you to Mr. Karthik, to Shravani, and the team of BioLeaks and APR for giving me an opportunity to be part of this summit and deliver my talk. Thank you so much for patiently listening, and sorry for the inconvenience being caused. Thank you so much. Over to you.
Thank you so much for the wonderful presentation, sir. It is very innovative and positive. Thank you. Thank you so much, sir.
Thank you. Thank you.
Next, I would like to welcome our keynote speaker, Dr. Sunita M. Reddy, ma'am, Associate Professor and HOD, Center of Pharmaceutical Science, Institute of Science and Technology, Jintu H, Hyderabad, India.
Yeah, hello. Hello, Ma. Yeah, hello, Salahuddin. How are you?
Hello, hello. Good morning, ma'am. Good morning. How are you? So nice to see you. Your voice is not audible.
Okay, let me check 1 second. Can you hear now?
Yeah, yeah.
Okay.
Yes, ma'am.
Please give me the share option, Amma.
Yes, ma'am. Yes, ma'am.
So, how you have been, Salahuddin?
I'm doing fine, ma'am. So it's been long time. Hope to meet you again physically at Apticon. But thanks to BioLeaks for making us to interact virtually.
Yeah, I don't think I from
Madam,
tomorrow.
your voice is missing. Probably there must be some audio.
Can you hear me now?
Yeah, yeah.
Can you hear me? Okay. I'm not coming to Opticon because so much work is lined up from tomorrow onwards to up to 5th. Because all affiliation work is there. So I thought of doing in a last minute to come and see everyone, but I don't think I can make it.
Yeah, yeah. Hope to meet you there at JNU again.
Yeah, sure, we will do that.
Make Sunita Madam co-host so that she can share her slides.
Yeah, I'm sharing it. Whether is it visible or not?
Yes, ma'am. It is visible. It has started sharing.
Yeah, I did. So
Yeah, it is visible, ma'am.
yeah. Okay, can I go ahead and start? Or any
Yes, ma'am. Please.
anything? Okay. Thank you everyone, and thank you BioLeaks and Pharma Summit, and we have associated for international conference also last year. Hopefully, we'll do it in again 2023 also. Okay.
Ma'am, sorry for inconvenience, ma'am. Your voice is not audible.
Okay. 1 second. Hold on. 1 second. Let me put my full voice. Okay, is it visible now? I mean, audible?
Yeah, it is
Yeah,
audible,
yeah.
but yeah.
Yeah. So, and you can see my screen also. Yes?
Make it to slideshow mode.
Yeah. So, um, is it audible? Is it visible, my screen?
Ma'am, screen is visible, but it is not on the slideshow mode.
It is on slideshow mode. I kept it on slideshow mode only.
There must be some technical issue
Yeah,
probably.
I
You can
kept
go
it on
ahead.
slide mode only. Slideshow mode only.
Madam, you exit and go with presentation
Yeah,
individually.
I'll stop sharing and again I'll share it. 1 second, okay? One second. Is it visible, my slide? Hello?
No,
Yes,
madam.
ma'am.
Is it visible? No? Yes, sir.
Ma'am, it is visible, but
Ah,
it's, yeah. Okay.
is it visible?
Yes,
Okay.
ma'am. Yes, ma'am. It is visible.
Okay. I'll put it in full screen now. Okay. So I'm going to start on lipid nanotechnology. This lipid nanotechnology is one of the novel drug delivery systems. Okay. So this novel drug delivery systems, we usually we have a we prepare so many formulations and so many carrier systems. But most I'm going to talk is only about the lipid-based carrier systems. So lipid-based carrier systems, there are several of different ones are there. One is like liposomes and neosomes. We are talking about nanoemulsions and solid lipid nanoparticles and different applications of lipid-based nanoparticles I'm going to talk today. And what we are going to talk, I mean, what I'm going to explain today is about this lipid major biomolecules. Okay, these biomolecules made up of like a polar groups and non-polar groups. Like most of them are polar groups. These polar groups are made up of usually like fatty acids, phospholipids, glycerols, and triglycerides. And these liposomes they have a head and tail. This tail is made up of again fatty acid chains, and whereas the head is made up of polar groups and they are linked with the different phosphate groups. So what is the advantage of this grouping in the liposomes or phospholipid molecules? Because you can have a polar regions and non-polar regions. So usually when the drugs are hydrophilic in nature, you can combine with them in the polar group. When the drugs are lipophilic and they're hydrophobic, then you can combine them into the tail parts, okay, and non-lipid part. So they have several advantages because in a single a lipid molecule, you can have two different drugs, and two different drugs can be transported to the body tissues, and you can incorporate more than one drug in one of the dosage forms. Okay, that is the advantage of these all lipid-based drug delivery systems. So you, when you see this cross section of all these lipid-based drug delivery, this is the liposome, which is arranged and the head and tail, and you can see that there is a hydrophilic molecules, and in the tail you can have a hydrophobic molecules there, and this is a solid lipid nanoparticle, and solid lipid nanoparticle you can keep the drug in a solid in inside of the aqueous layer, whereas you can have a another particles are another lipid particles on their heads, and there is you can have a functionalization on the top of that. You can add some other functionalization groups or functional groups where you can transport this medication to the actual target tissue. Okay, and other one, it is a nano emulsions. These nano emulsions, again, you know that there are dispersed systems, and they will be like a oil in water or water in oil, and in a oil you can keep the lipid soluble drugs, whereas the water you can keep the water soluble drugs. and both can be kept it in a oil and water emulsions. Same thing like a miscellaneous. Miscellaneous also same concept, but usually we can usually in the research we can keep only one type of drugs in a miscellaneous. We can encapsulate. Okay, and what are why they are usually useful in a pharmaceutical carriers because these are all carriers you can give as IV administration. Because whenever we talk about IV administration, the drug reaching into the systemic circulation is 100%, and 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 therapeutic levels will be reaching very high, and 100% of drug is available for the treatment. And all these lipid nanoparticles are biocompatible, biodegradable. And why they are biocompatible? Because they are made up with fatty acids and lipophilic molecules. So these are all biocompatible with all our tissues and also our muscles, because most of them are made up of lipid layers. And biodegradable because these are fatty acids, and very easily they can biodegrade. So there is no problem in metabolism also. Once the drug is released, whatever the carrier can be easily excreted to the body by biodegradation. Okay, and one more, there is a prolonged circulation. Once you are given in IV administration, you will have a prolonged circulation. Same molecule will be circulating several times and reaching into the tissue which is required, which is a treated, which is a target required. Okay, and they have a small size. When you know that when they are made up in a nano size, their surface area is very high, so you can have a high loading of the drug. If it's a high loading of the drug, with a one carrier you can carry the many molecules of the drug molecule, and much dose can be loaded into the one dosage form. That is another advantage of these lipid carriers. Another one is tumor accumulation. You know that whenever the tumors are present or cancer tissue is present, they are in leaky in state. The cells are very leaky. So, but when you are these carriers, the nano carriers, when you are given these molecules, either liposomes or neosomes or solid lipid nanoparticles or MSLRs, they will be accumulating into the tissue, those cancer tissues. So here you can see that there are so many layers that you can have. This is your drug molecule, drug crystallized in aqueous fluid, and on the surface of that the head. Hello, that is a head portion.
Ma'am, sorry
Yes,
for the inconvenience. Sorry to interrupt you, ma'am, but your slides are not moving, ma'am. It's still on the
not
second
moving.
one.
Okay, I'm not sure why it is happening. Let me. Okay, shall I go ahead and share again? One second. Okay. One second. Can you see now? Hello? Can you see the slides now?
Ma'am, we can see the slides, but they're not scrolling down. Yeah.
Let me through now. What about now? Are you able to see the sixth slide?
No, ma'am.
Yes, happening.
Ma'am, you can share us your PPT to us. We will share for you.
Yeah, now liposomes it's coming.
Yeah, you can see
It's
the
moving.
liposomes. Yeah, you can see now.
Yeah,
Yes,
it
ma'am.
is.
Yeah, so this is the slide I was in.
Thank you, ma'am.
Yeah, like the carriers, they are very advantageous because they have a lot of advantages when you given in a IV administration. Okay, and now I change the slide. So can you see the slide now? Change its slide.
Yes, ma'am. Yes,
Yes.
ma'am.
Okay. So what are the liposomes? These are the smallest round structures and produced by natural non-toxic phospholipids and cholesterols. And you can load with so many huge varieties of molecules, and you can have like a small drug molecules, proteins and nucleotides, even plasmids and lipid particles can be loaded to these liposomes. Okay, and these are the advantage of these liposomes is that you can have a hydrophobic and hydrophilic drugs in one single molecule of these liposomes. For example, you have a liposome cisplatins, lipoplatins, anti-cancer drugs, and they are composed of natural lipids like phospholipids and cholesterols, and a very lower risk of toxicity because they are very biocompatible and biodegradable. And you can include different materials as a tax, and like you can have antibodies, antigens and cell receptors, nucleic acid, all these can be added to the surface of these liposomes. So these liposomes can carry more than one drug and more than one molecule. And the size is approximately about 50 to 800 nanometers. When 50 to 800 nanometers, and the circulation of these products will be very high in our blood circulation, and recirculation also can be done. Okay, and these are all these liposomes. You can talk about the conventional liposomes, stealth liposomes, or targeted liposomes, or cationic liposomes. Why they are different? Because a conventional liposome will not have anything on the surface molecule. Okay, but a stealth liposomes, you can have a pegylated liposomes. You can have a polyethylene glycol molecule attached onto the surface of the liposomes. When you added a pegylated, the enhanced permission effects will be there, and they will not be recognized by the reticuloendothelium system. And when they are not recognized by endothelium reticular system, the circulation of liposomes will be many times. So it will be, then the drug molecule will be entering into the tumor. And another one is a targeted. Targeted means you want to have a molecule which has to be targeted to certain receptors. So these targeted molecules which will be attached in the surface, similar to the receptor which are available in the tissue. So these targeted will go to the exactly that location and target that receptor, so the action can be initiated. Another one is a cationic. The surface will be a cationic liposomes. These cationic liposomes are advantage because they will have a positive charge, and they will go and attach to the negative charge molecules, and they will have an action. So in a liposomes, you can have like a conventional, stealth, targeted, or cationic, depending on what you want to do or where you want to target them. Okay, and these liposomes are different types, that's multilamellar vesicles, large unilamellar vesicles, small unilamellar vesicles. These multilamellar vesicles will be having more than one ring, and they are a size of 0.05 to 10 micrometers. And also large unilamellar molecules, they will have a one single layer, and they are made up of 50 to 100 nanometers. And small unilamellar vesicles, they are very small in size, 25 to 50 nanometers, and they will have only one single phospholipid layer. Okay, yeah, here you can see that small unilamellar vehicle 25 to 50 nanometers. This is a large unilamellar about more than 100 nanometers. Multilamellar, so many layers of phospholipids will be there. So once you are increasing the layers, the drug molecule incorporation into these liposomes can be increased, and drug loading also can be increased, and multiple drugs can be incorporated into the one liposome. Okay, one type of liposome. So that is advantage of this. Okay, and the same thing here, this is a same diagram. which is showing how the actual one lipid bilayer will be lay, and how the small unilamellar and multilamellar vehicles will be present, and how you can prepare them. So once they are this given, or they are given as IV administration, they have to enter into the cells. So then once they are entered into the cells, there are so many different ways of entering to the cells. One is a endosomal method. One is endosomal method. They will be directly entering, and once they enter into the cell, the surface area will be detached, and the positive sensitive liposomes will be entered into the actual cell molecule, and some other liposomal material, they will be entered through the pinocytosis, and once they enter into pinocytosis, lysosomes will be acting, and the actual drug molecule will be releasing, and some cationic liposomes, they will be directly crossing with the intracellular membranes, and the DNA will be detached from that, and they will directly enter into the nucleus. So there are so many cellular uptakes can be done by the liposomes, and that way the drug concentration can be more into the actual affected cell. Okay. And retention of both lipophilic and hydrophilic drugs is one of the advantage of this. And these are all like, you can have an antibody or ligand can be conjugated onto them, and they are very very minimum of antigenicity. There will be no reaction once they are given, and as I told, they are very biodegradable and biocompatible. And liposomes can be prepared by three different methods. One is a rehydration vesicle method, and this is developed by Kirby and Gregoriasis. He's two of the two scientists in 1984, and they made a small unilamellar liposomes. What they did is they are lyophilized, the freeze dried. What they have done, they have taken the liposome, the empty liposome, and they have dispersed the actual drug which need to be entrapped. And as soon as they disperse, they have done the freeze dry. Whenever you have to inject them, whenever you have to do the IV administration, you have to take the small volumes of the water, which is a sterilized water, and add them and redisperse them and immediately administer to the patient. Okay. And this is a very simple method, very simple, mild conditions are required, and very high encapsulation efficiency is there. And this we can do it in our lab also. In a JNTU, we have been doing this several drugs we have been doing in the liposomal formulation. Okay. And this is the method how you can do. This is your empty small unilamellar vesicles, and you mix with the same amount, same portion of the drug, which is equimolar concentration. Then immediately you freeze dry them. Once you freeze dry, they become stable, and you keep them. And whenever you want to re-administer, you want to administer to the patient, and hydrate with them 1 by 10 of the initial, means 1 parts of your freeze dried products, and also 10 parts of your liquid water for injection, and rehydrate them and administer to the patients. Okay. So other one is the detergent removal method. Detergent removal method is like you mix with a lipid detergent micellars, and which will end up in large unilamellar vesicles. So when the large unilamellar vesicles are done, and you do the freeze thaw sonication methods. So you repeat the cycles, like you keep them in the freezer and thaw them. You keep them and thaw them, and meanwhile you add the actual molecule which you want to be encapsulated. Once you need to whatever you want to be encapsulated, because of repeated freeze thaw methods, that end up in lot high efficiency of the encapsulation of the drugs. Okay. And one, the most important is the we can do the techniques based on a different after preparation, either by this freeze dried method or sonication method. You need to do the purification, because lot of other lot of other material will be present. That material has to be taken out. That you can do is a centrifugation technique or dialysis technique or gel filtration column chromatography technique also can be used for the purification of these liposomes. This is the centrifugation method, nothing but you do the centrifugation, with a high amount of buffer need to be added and centrifuged about 15,000 rpm for about 20 minutes at a room temperature, and decant your substance. Only small amount of liposomes which will be gathered that can be collected, and repeat this process several times until you see there is no color at all. So that indicating your pure liposomes has been formed. These liposomes you can freeze dry and store them. Another one is a dialysis method. Whatever the liposomes you have prepared, again add excess amount of buffer and dialysis. Keep on shake them for so many hours and repeat so many times until you see new color in the dialysis membrane. Okay, and the color can be detected by fluorescent detectors. Whether any color present or not, that indicates the purified liposomes. Okay. And another one is a column chromatography. This chromatography we can also use to purify these liposomes. What we do, we take the Cephadex, the polydextran beads, and we pass these our liposomes through these beads. Then usually whatever the molecular weight of this Cephadex we take is about thousand less than 1,000 Daltons. Okay, the size is less than 1,000 Daltons, and the molecular weight is less than 1,000 Daltons. And you pass this liposome through this Cephadex Z50 and collect the end of the your column chromatography. Usually they are purified with all other material is taken out. But there is a possibility, there is some disadvantage of this material, this method, because always we expect a low yield in case if it is a the Cephadex Z50 has a different size. Okay, because of the different size, sometimes we expect very low yield, but you can always modify the method, and you can modify the Cephadex size also. Then you can get the high yield, or depending on which liposomes you are trying to purify. Okay, usually we prefer medium or coarse grades of Cephadex, the particle size about 50 to 150 micrometers, and that will give us a very good multi-layered vesicles, or small unilamellar vesicles. They will be purified very easily. Okay. And next one, lipid-based drug delivery system is neosomes. These neosomes looks like same as a liposomes, but only we are using a non-ionic surfactant. Okay, and this is an alternative method for the actual liposomes, because whenever we do the liposomes in our lab and all this, the problem is stability, because unless we freeze dry them and make a stable liposomes, if you leave them in the room temperature for a prolonged period, there is always a problem. Separation of the liposomes can be occurred, and denaturation of the liposomes are occurring. So this is an alternative method for the liposomes, and they are look same as a liposome. They look same as a liposome, but the stability is very high. The stability problem which we experienced in the liposomes can be overcome by the neosomes. Okay, and they also prepared in the same way. They are also available in a unilamellar or multilamellar structures, and they also you can keep the both lipophilic and also hydrophilic drugs. But the cost of these preparations are very low when you compare with the neosomes. Okay, and not only neosomes, we are using non-ionic surfactants in our formulation. Once you are using a non-ionic surfactant, the penetration of the drug is become enhanced. When the penetration of the drug is enhanced, for in case of like a cancer, all these tumors, the drug accumulation into the cells will be very high. So the therapeutic effect you can see that. Another one is a miscellaneous. Miscellaneous is like a single tail structures. They look like a same as a liposome, but we can incorporate only one drug, unlike a liposome or a neosome. In neosomes and liposome, we can have two different drugs. One is in head and one is in tail. But here it is not possible. We can do usually only one drug. Okay. Other one is a nano emulsions. You know the nano emulsions. This has been in the market for several years. The emulsions are in the market for several years, but nano emulsions are coming into picture for only recent in a novel technology. because these why they are not very popular, because they are thermodynamically unstable, and the stability has to be enhanced by one of the surfactants. But this is a nanoemulsions. When you are able to keep practically insoluble drug to the soluble emulsion, the bioavailability of those nanoemulsions are like a 2 to 3 folds than a conventional emulsion. Okay, and this is the nanoemulsion. The self-emulsifying or self-nano-emulsifying drug delivery systems are like isotropic mixtures of surfactant, co-surfactants, and oils, and where you are keeping practically insoluble drugs to the soluble emulsion form and a single solution form. These emulsions become these solutions become emulsions with a moderate shaking. That is advantage of emulsions, and you can keep variety of drugs in these emulsions. Okay. Okay. Another most popular one is solid lipid nanoparticles. Because so far we spoke about liposomes, neosomes, and emulsions, but sometimes we have to keep the drug in a liquid form. But here, solid lipid nanoparticles, we have to keep the drug in a solid form. We can keep the drug as it is, but we are making them into the small nanomaterial and stabilized by the surfactants. Okay, and they are very spherical and shape and same the advantages they have. And the core material that we are making is the fatty acids or acyl glycerols, waxes, mixture of these surfactants. All these can be used. Okay, and biological membranes which we are using for solid lipid nanoparticles are like phospholipids, sphingomyelins, bile salts, and sterols, and all of them we utilize as a stabilizer. Okay, and the advantage of this is there are very minimum carrier cytotoxin. Okay, that is a more advantage. Okay, and these are the solid lipid nanoparticles. You can see that here is the drug that you have solid in a lipid solid in a lipid layer, and also phospholipid molecules are surrounding them, and they are surface charged by pegylated or a polyethylene glycol. That way, the stability of these solid lipid nanoparticles is enhanced, and you can have a prolonged therapy or recirculation of the therapy can be achieved. Okay. For example, you can see here, you can have a positive charge drug and also negative charge surfactant, and combine them and make it as a solid lipid nanoparticle and stabilize them by you can stabilize the surfactant, and you end up in a solid lipid nanoparticle. And this has a more advantages when you compare with the others. Okay, and this is the same morphology. You can have that. Whereas in solid lipid nanoparticle, they are again modified into lipid polymer nanoparticles. Lipid polymer nanoparticle, whereas a lipid polymer nanoparticles, where you have a PLGA as a polymer and drug molecules are entrapped as in a matrix. Okay, and again they are stabilized by the surfactant and surface charged by the pegylation, and these are more stable and more receptive. Okay, they are also called stet layers. Pegylation has done to prevent the destruction by the reticuloendothelium system. Okay, and how they are prepared? They can be prepared by top down method or bottom up. The top down method means high pressure homogenization. You take a big particle, make it into the small ones, and microfluidization method or membrane contractor. Or you can take the bottom up technology, emulsification solvent evaporation method, emulsification solvent diffusion method. Both can be adopted, that you take a small lipid molecules, make it into the solid lipid nanoparticles. In both way, you can incorporate the medication or drugs into the molecule. Okay? So what is the advantage when you compare with this? This is your liposome. Okay, this is your liposome. This is lipid nanoparticle or nanomulsion. This is the lipid nanoparticle. The liposome, lipid bilayer enclosing an aqueous core. This is the aqueous core. You can have a several hydrophilic drugs in here. When you come to the nano emulsion, you are making it the oil versus oil into water. Then you are keeping a lipid drug into the layer. Lipid monolayer is enclosed. Whereas when you compare to the lipid nanoparticles, lipid monolayer enclosing the solid drug is your keeping. So this is more stable when you compare with these two. Okay, that is the advantage of this solid lipid nanoparticles. And each and everything, all lipid dosage form, they have their own advantages, own way they can carry the medication. But this is a comparison between the three of them, which we discussed now. And like lipid emulsions, there is some kind of disadvantages of all these materials. Lipid emulsions, they will have a drug leakage. Also, there is the possibility of hydrolysis, and you can lose some drug. And particle growth is also can be seen unstable during the storage. And the same thing with the liposomes, they are unstable during the during the storage, and the particle growth is also possible. Drug leakage is also possible. But when you compare it to the solid lipid nanoparticle, these are all disadvantages are overcome. No drug leakage due to solid matrix are stable against the hydrolysis of drug and against the particle growth. And drug release is for a prolonged period of time. And comparatively, they are stable because the drug is in the solid form and you are keeping them into the lipids. Okay. So how they deliver? All these medications usually be delivered by the intravenous, intramuscular, or subcutaneous, and their size is less than 1 micrometer. So the systemic distribution is very, very high, and there is no blood clotting can be seen, and there are sustained drug release also can be done, and the incorporation of drug is gradually released by degradation of by enzymes or by diffusion of the particles. The rate of release may be controlled by the nature of the lipid material or a particle size or a surfactant or inner structure of solid lipid nanoparticles. So there is always there is a advantage of this solid lipid nanoparticles. Okay, but overall lipid drug delivery, has several advantages than a conventional, because the conventional will have a release the drug very quick and degrade very quick, but it is not in the case of lipid nanoparticles. Okay, and the advantage of lipid emulsions or polymeric nanoparticles, they have a overcome the temporal or in vivo stability issues that always will be seen in case of conventional material. Okay, and allow the incorporation of lipophilic and hydrophilic drugs in the same molecule, and they are non-biotoxic. Okay, and enable the possibility of controlled drug delivery or drug targeting can be possible. And more is increased drug stability and no problems in preparation of large scale. So that is the overall advantages of lipid-based drug delivery systems. And by seeing the lipid drug delivery systems of liposomes, neosomes, solid lipid nanoparticles, and one more new carrier has been developed, that is called nanostructured lipid carriers. Nanostructured lipid carriers are little different between solid lipid nanoparticles. That is, they are made up of matrix structures. Okay, they are made up of matrix structures when you compare with a solid lipid nanoparticles. Okay, and these solid lipid nanoparticles are somewhat superior than nanostructured lipid carriers are little bit superior than solid lipid nanoparticles, because they can be used in lot of cosmetic applications. Okay, and here you can see that this is our solid lipid nanoparticle, whereas the nanostructured lipid carrier. So this lipid carrier is more advantage because uh, um, uh, the actual carrying of more drug molecules is possible in nanostructured lipid carriers. Okay, and this all nanotechnology. How that this nanotechnology, lipid nanotechnology, is useful, that nano liposomes, nano neosomes are used for cosmetic industry as a delivery of the different vehicle. And solid lipid nanoparticles, nanostructured lipid carriers has a better performer than a liposomes. And nanolipid carriers have been identified as the potential next generation cosmetic drug delivery systems. Like they can provide the skin hydration, bioavailability, stability of the agent, and control occlusion also possible. Okay, so they have a different routes of administration. They can have a transdermal or topical application. They can have intravenous administration. They can have a oral administration, they can have a intranasal administration. They also like they can use it in the drug screening microarrays method, drug delivery system, and also ocular drug delivery. They can use vehicles for vaccination. They can use, and they can use in a cosmetic. I'm going to tell one example each, and I'm going to start it. Okay. This is a solid lipid nanoparticles. You can use it in a microarray method, screening like a diagnostic purpose. We can use what is that we do? Surface mediated delivery of drugs to cells from microarrays is possible, and it can be delivered by multi-layer phospholipids, and each layer can be delivered, or each layer can be eroded to deliver the medication on the surface. Okay. And other one is this is solid nano particle can also deliver the drugs to the via cell plasma membrane. Okay, into the cell via plasma membrane, and they can be intracellular or intracellular delivery can be done. And liposomes in a cancer therapy, that is one of the most targeting drug delivery that can be used, and that can be done in a several drugs. Okay, several drugs can be done. And also gene therapy also we can initiate with this all the solid lipid nanoparticles or a lipid based drug delivery systems. Also gene delivery also can be done. Okay. And they also can do in ophthalmic drug delivery. These are all ophthalmic drug delivery because usually ocular bioavailability can be enhanced by these hydrophilic and lipophilic drugs, and usually we required whenever we are doing ophthalmic drug delivery, we need a sterilization of the product. That can be possible. And also these lipid based drug delivery systems are also for the carriers for several vaccines. Now you have seen there are so many COVID vaccines has come, and most of these lipid carriers as used in the vehicle for delivery of the this vaccine. Okay. And topical drug that they can be done. Topical drugs advantage of lipid nanoparticles. They have several advantages on a topical drug delivery because increasing the drug permission to the stratum corneum and lower the skin irritation, and also extend the effective time. They will have a retention of the medication on the area where you apply, and due to increased retention on the skin, the amount of drugs that entering into the epidermis and dermis is become more, and systemic circulation and mitigation of toxicity will not be there because they are in the surface. And phytophobic drugs that liposome formulation increases the overall solubility without use of any irritatable solvent. That is another advantage of this lipid drug delivery system. Okay. And the moreover the hello, hello. Okay, hello, hello, can you hear me? Yeah, can you hear me? Yes. Hello, hello. So the lipid nanoparticles are patent products of like a worldwide lipopearls or nanopearls is the one of the systems which are more into the market now. Okay. And there are a lot of benefits in using the lipid nanoparticles in cosmetics. Um, okay. And one more, the use of this nanomaterial is like, whenever you have like, sun tanning. Because sometimes whenever we go in a sun, there is a UV radiation produced as the TT diamonds. Okay, these TT dimers end up into the tanning and discoloration. This is because of T4N5 enzyme will be repaired, like a DNA repair enzyme. T4 endonuclease. T4N5 is a 16 kD protein, and these need to repair DNA damage. Long-term skin metabolism and UV radiation causes the thymine bases in a DNA to form a lesions like TT dimers. Okay, the T4N5 when we make is a nanomaterial that can replace the TT dimer and allow the DNA replication normally and resulted in the healthy skin conditions. That is another advantage of this lipid-based drug delivery systems. And they can be used for different targeting. They can use it for the respiratory targeting. They can use for the brain targeting. They can use for the ophthalmic targeting. They can use for the cancer targeting. These are all can be advantages of this materials. And that I'll stop my slides. And I think that is the time is done. Hello, anybody there? Hello, Savani? Hello.
Hello, madam, Sunita, madam.
Yeah.
Yeah, thank you, madam. It is very nice lecture, madam.
Yes, I am here.
Yeah, it's very nice lecture, madam. I'm Dr. Ramana, Siddhartha Pharmacy College, Vijayawada, madam.
Oh, nice meeting you.
Yeah.
Yeah, looks like the organizers. I don't know whether they are there or not. I'm not sure. Okay.
Thank you so much, ma'am, for the wonderful lecture. Thank you, ma'am.
Thank you, ma. Thank you. Okay, so I can exit. Is it Dia? Who is that?
Thank you, ma'am.
Okay.
I'd like to welcome our keynote speaker, Igna Pulis Lara sir, Board of Directors from IQ Bold Ventures, Spain. Sorry, due to some technical issues, sir will be not available. I would like to welcome our next keynote speaker, Mrs. Priyama Vada Jain, ma'am, Associate Director from Jubilant Life Science Limited, Delhi, India.
Hello.
Good morning, ma'am.
Hello, good morning, everyone. Am I audible to you?
Yes, ma'am, you're audible.
So let me introduce myself once again. Good morning, everyone. My name is Priyambada Jain. I work in regulatory affairs in a pharmaceutical company. I work with Jubilant Life Sciences. So if it is possible for you, can I have the introduction of the participant, just the name, so that it is easier for me to understand the kind of audience I'm speaking to?
Just a moment, ma'am.
Oh, okay. I can see all the name on the screen. So we have Nidhi, we have Yashwini. Perfect. So I would not like to present anything in a presentation mode, or will show you a presentation. Rather, I would like to have it a very, very interactive session. And I would like you to please interact, ask questions, participate in the discussion rather than having a one-way communication. Is it okay for all of you?
Yes, it's fine.
Okay, perfect. So before I start to say whatever I wish to say,
I request Pallavi ma'am to please mute yourself. Sorry for inconvenience, ma'am.
No, it's perfectly fine. I do understand the Zoom meetings and technology has its own pros and cons. That's perfectly fine. So since my domain is regulatory affairs, I would like to know from the participant, maybe you can just raise hands or you can just type in a comment. What is your understanding of regulatory affairs? If you'll see, each and every pharmaceutical company is having a regulatory affairs department. And these are the guys who are doing something. Probably, what is your understanding? What these people are doing in a pharmaceutical company? Why this department is at all required? Okay, perfect, Kripa. Thank you so much. Spoke between the agencies and the manufacturer. Perfect. Anything else you would like to add? Any other thought from anyone else?
Certainly to maintain the quality of those products, and also as per the regulatory affairs rules, it is different from different countries. So as if you are sending the products in different countries, so we have to maintain as per their rules and regulations. So it is very much required if you are sending your products in different countries. So that's why it is one of the most important department of any pharmaceutical company.
Yeah, thank you so much, Samrat, for adding that to it. So yeah, as Samrat said, and as Kripa also said, Regulatory Affairs Department. Yeah, thank you. Thank you so much. Definitely, Kripa. So we know that Regulatory Affairs Department, it acts as a bridge between the regulators. When I say regulator, that means regulatory agencies and the pharmaceutical companies, because it is very, very important for each and every drug product to get authorized or get registered in the country, in whatever country we would like to sell this product. So we all know this, but do we also know the background, why it all has started, from where it has come from? You know, I feel that nowadays we have a lot of current knowledge about many things. We know why regulatory affairs is there. We know about innovations, new technologies, but are we also rooted to our roots? Why and how it all has started? So rather than talking about what is new, because what is new is already there in the public domain, and a lot of eyes are there already. Okay, what is new is coming in, what we are doing now, but are we also know about our roots? If I belong to a regulatory affairs department, do we also know why it all has started? What was the need? So for this, I'll take you back in time and let me tell you a story. The story goes like this. Good morning, Nidhi. So the story goes like this. The year was 1901 and the place was St. Louis, America. And St. Louis, America was under the epidemic of diphtheria. As you all know that diphtheria antitoxin is prepared by using horse serum. So it was prepared by using horse serum. And there was a horse. His name was Jim. He got infected with tetanus. Now what happens is this tetanus contaminated vaccine, or sorry, antitoxin was administered to people, which led to the death of 13 kids in St. Louis, America. A similar shocking incident also happened in Camden, New Jersey, where the smallpox vaccine was given to kids, which was again contaminated with tetanus, led to the death of 9 kids. So because of these mishappenings, the Biological Control Act was passed in 1902 in America. So by this act, government got some control over how the biological products are manufactured. So they got to inspect the facilities where these products are manufactured. They exercise control over these products. So, and you know what happens? When some tragedy happens, some law is passed, we feel, okay, now everything is okay. We are very, very safe. Nothing will happen now. But then there was a chemist. His name was Dr. Harvey Valley, who was doing a study in America again. He was doing a study on 12 volunteers, and he named this group as a poison squad. And what was going on in this poison squad? Dr. Valley, he administered food or given food which was laced with colors and preservative to these 12 volunteers. And he called these 12 volunteers as a poison squad. So this work was going on between 1902 to 1906. And at the same time, there was a journalist by the name of Upton Sinclair. He also published a book by the name of The Jungle. The Jungle book was about the lives of immigrants who were living in America and the kind of insanitary practices or the life they were having in America. So when these two works were published in 1906, people got to know not only about the insanitary or unsanitary practice, living conditions of immigrants, but also the book also had the shocking disclosures of unsanitary practices in the meat industry. When both of these work came to light, came to public eye, they led to public uproar. And because of this public uproar, in 1906, 2 acts were passed. One was the Meat Inspection Act, and another was the Pure Food and Drug Act. So let's try to understand what was Pure Food and Drug Act at that time. So Pure Food and Drug Act was emphasizing only on the labeling of the product. What it used to say that whatever ingredients, the active ingredients you have in a drug product, if it is potentially harmful, if it is addictive, it should be clearly labeled or put on the label in the quantity in which it is present. It also required the ingredients which are present in a drug to meet the pharmacopoeial standards with respect to purity. And it also prohibited the interstate trade of adulterated medicines. And adulterated at that time meant only the labeling. If you have put in something in the drug, you should put it on the label also. That was all it said. As far as food is concerned, there was no analogous standard for the food. Anyone has any question at this point in time? Okay, then let me continue. So, though there were some standards for drugs, but there were no standards for Okay, then let me continue. So though there were the standard for the drugs, but there were no standards for the food as such. The only thing food required to have is that it should not contain anything which can be called as food replacement to food. And it should not contain any material which is stale, which may be harmful, but no regulatory standards as such. Then what happened in 1911, there was an inventor who was an entrepreneur also. He came up with a medicine for the treatment of cancer, which he called as mild combination therapy for cancer. At the same time, the US government, when they were putting in control, like the Biological Control Act, the Pure Food and Drug Act, and the Meat Inspection Act, they were also inspecting the various manufacturer. And if something is not found as per the standards set by the government, they were also putting fines on them. They were doing confiscations, but only the small confiscations were happening. When they raid this inventor and entrepreneur, Dr. Johnson, so he did not fall in line with the government's ruling. He said, okay, no, whatever fine you're imposing on me is not acceptable to me. And he took this straight to the court. And court in 1912, rather 11, he gave a ruling that the false and misleading therapeutic claim does not fall in line with the Pure Food and Drug Act, as it does not talk about that you cannot put false therapeutic claims on the label. To overcome this ruling and again to protect the public health, Shirley Amendment was passed in 1912. Okay, so till now, by 1912, we had the Pure Biological Control Act, we had Pure Food and Drug Act, we had Meat Inspection Act also. And then the Shirley Amendment was also done in 1912. But till now, nobody was talking about the safety of the drugs. And efficacy was way far off. You can just combine anything, give it to the patient will take it. Whether he's getting benefited from it or not, it was nobody's concern. Again, safety was also not assessed. So what happened? Because of these reasons, these were the disadvantages, or these were the, you can say, the loopholes of the law. Because of this, many other tragedies also happened. Like, there was a product by the name of Banbur, which was the worthless cure for diabetes. There was Wilhite, which falsely claimed to be treatment for tuberculosis and other pulmonary diseases. Then there was the Lashlior tragedy. Lashlior was an eyelash dye, which caused severe damage to the eye of many females in USA. And there is a one confirmed case of permanent blindness also because of Lashlior. Then there was Mrs. Willow's soothing syrup. The soothing syrup was for the colic and the teething babies. But then it was laced with morphine and killed many infants across USA. So you can see that till now, drugs were there, many drugs were there. They were not safe and nobody knew about the efficacy at all. Then came your 1937 when sulfanilamide elixir tragedy happened. So there was a pharmaceutical company, S.E. Messon-Gillen Company. What they did, they dissolved sulfanilamide in diethylene glycol, which resulted in a fatal mixture. And it killed more than 100 people across USA. Because of these tragedies, the government ultimately thought that there is a need to have a law in place, which is ensuring that whatever products are going to the public use, they are safe also. So they came up with, in 1938, the Food, Drug and Cosmetic Act, जिसको हम आज जानते हैं as FDNC Act के नाम से. So that came way back in 1938 in USA. Now, when this act has come, within a span of 2 months, FDA started to recognize many drugs which cannot be regarded as safe to be used by the patient directly, like sulfur drugs, because they were having many side effects. So they started identifying these kind of a drug, and they thought that you cannot give any medicine just like that to the people, to the patient. There should be 2 categories of drug. The drug which can only be given under medical supervision, and the drug which can be given directly by the pharmacist without any medical prescription, which led to the 2 categories of drug: prescription drugs and over-the-counter drugs, which happened ultimately legally and by law, which happened in 1951 by Durham-Humphrey Amendment. Okay, then I'll continue. When USA was making laws, amending laws for the public benefit, a tragedy happened in Europe, Western Germany, due to thalidomide. Thalidomide, a drug which was available as an over-the-counter drug, as a sleeping pills, as well as for the morning sickness. And it led to phocomelia in Western Germany, and more than 10,000 kids or infants got affected due to thalidomide. Phocomelia is a condition where underdeveloped limbs and if you have any question, you can ask me. If you would like to add something, you can definitely add to it. If nothing is there, then I'll continue. Okay, let me continue. So because of this thalidomide tragedy, more than 10,000 infants got affected, and approximately 40% died at the time of birth. Till this time in Europe, there was no system of assessing the safety of the drugs. And this news of thalidomide tragedy also reached to USA, where the news was there regarding the role of FDA inspector, Dr. Francis, in keeping this drug off the US market. Since this drug was never approved in USA due to safety issues, US was safe during thalidomide. And because people got to know that Europe suffered due to thalidomide massively, they again requested the government to have stricter laws regarding the safety of the drugs and efficacy of the drug, which ultimately led to the Keffer-Harry Amendment in 1962. So till now, since 1938 to till now, till 1962, the emphasis was only on the safety of the drugs. But due to Keffer-Harris Amendment, the focus shifted only from safety to efficacy also. Now the drug manufacturers were required to prove that the drug is safe as well as efficacious before it is reaching the market. Since Europe was also shaken due to the thalidomide tragedy, they also put a system in place in 1965 that whatever drugs are going to the market for the public use, they should be first authorized through a formal system of market authorization. So this is how in 1938 it started in USA that a drug manufacturer needs to prove that the drug is safe to use. And then in 1962, they amended the law to also ensure that it is safe as well as efficacious. And Europe started the regulation, the formal formation of regulations in 1965, so that whatever drugs are reaching to the patient, they are safe and efficacious. Before this time, there was no such laws that your drug has to be proven safe and efficacious before it is reaching the market. Now, what happened to the products which were already there in the market since 1938, which were proved to be safe? So the government also asked for a retrospective analysis. Okay, we know that drugs are safe, but then manufacturers have to prove that they are efficacious also. And it led to the retrospective analysis for all the drugs which were approved in USA between 1938 to 1962. So this is the entire background of regulatory affairs and why we even today we see that the most stringent laws we have in USA and also in European Union. And later on, it led to the formation of ICH also, which is a tripartite committee, which include US, Europe, and Japan. But still, since they started early evaluating their drugs with respect to safety, efficacy, that's why they are very, very ahead of time when it comes to the coming up with new regulations, new norms, new guidelines. If you have any question, this is all from my side because I wanted to emphasize that we all know what is happening now, but we should also know how it all has started. What is the background? which unfortunately I saw missing while evaluating people for interviews. They know everything academically. They also know, okay, authorization is required, registration is required, but why it is required? What is the relevance? It is very, very relevant. The role of each and every pharmaceutical professional is very, very important to ensure that whatever we are doing, it is directly related to the journal public. So we have to do it in the best possible manner, keeping in the end user in mind. So this was all from my side. Thank you so much. If you have any questions, you're most welcome to ask.
Ma'am, I have one question.
Yes, please.
Not exactly related with regulatory, but being a teacher of an university, few of my students, they are very much interested to join regulatory affairs department. So what are the basic criteria which is applicable for a fresher to apply for the regulatory affairs department? Is there any specific specialization is required, or M.Pharm or B.Pharm degree will be enough to apply for the regulatory affairs department?
So B.Pharm degree is not enough. A person has to have a master's degree in any stream of pharmaceutical sciences. But nowadays, a lot of universities are having specific degree, master's degree in regulatory affairs as well. But through my practical experience, what I have seen is that any normal postgraduate person who is in postgraduate in any stream of pharmaceutical sciences is no different from somebody who is doing the master's in regulatory affairs, because as I said that the conceptual clarity is not there even in the people who are coming up as a master's in regulatory affairs. So anybody has done a master's degree in pharmacy can join regulatory affairs.
Thank you, ma'am, for enlightening us. Thank you.
Okay, thank you so much for being the wonderful audience. Have a good day.
Thank you so much, ma'am, for the wonderful interaction. Thank you, ma'am. Thank you.
Thank you, Divya. Thanks a lot. Bye-bye.
Bye, ma'am. I would like to welcome our session chair, Dr. K. Karthikarian Sir, Professor and Head School of Pharmaceutical Science, Wells Institute of Science, Technology and Advanced Study, Chennai, India. And I would like to welcome Dr. Priya Madhavan ma'am, Associate Professor from Taylor's University, Microbiology, School of Medicine, India. Dr. Tenny Thomas Jugnat Sir, Professor, Chemist, College of Pharmaceutical Science and Research, Department of Pharmaceutical Chemistry and Analysis, India. I request all the participants to join the breakout rooms. Hall. We'll be starting with a session 1. I would like to welcome our first presenter, Himani Dumka ma'am from Sardar Bhagwan Singh University, e-poster presentation on evaluation of hepatoprotective activity of polyherbal formulation against CCL4 induced hepatic damages in rats. Himani ma'am, are you present there?
Yes, ma'am.
Yeah. There is a small announcement for the participants. Your presentation will be of 8 minutes and 2 minutes will be of questionnaire. Total 10 minutes. Ma'am, please put it on the slideshow. It would be better. Yeah. Thank you.
Ma'am, am I audible?
Yes, yes, you're audible.
Good morning, one and all present here. My name is Himani Dumka. I am from Sardar Bhagwan Singh University, Dehradun, Balawala, Uttarakhand, India. I am giving a poster presentation on the topic Evaluation of Hepatoprotective Activity of Polyherbal Formulation against CCL4-induced hepatic damage in rats. First, we comes to the introduction. About the liver. Liver is an organ of paramount importance which regulates various physiological processes in the body, such as metabolism, excretion, and storage. CCl4 means carbon tetrachloride convert into CCl3 and CCl3OO free radicals in the presence of enzyme cytochrome 2E1. These free radicals then activate the inflammatory mediators, causes liver fibrosis, which are responsible for the liver injury. Alcoholic liver disease, primary liver cancer, cirrhosis, cyst, fatty liver diseases, jaundice, hepatitis are the type of liver diseases. And now we comes to the literature review. Polyherbal formulations contain several phytochemicals which may target several signaling pathways at the same time and may bring about benefit the synergistic and additive action. Ageratum, conjoids, aglamarmelos, and Tinospora cordifolia have also hepatoprotective activity. This study is focused to prepare polyherbal formulation of these three ethanolic plant extract by mixing them in different ratio. Now we comes to the method, how we prepare the polyherbal formulation in different ratio. First, polyherbal formulation 1. In this formulation, we take Ageratum conizole in 1 ratio, aglamarmelos in 1 ratio, and Tinospora cordifolia in 1 ratio. Polyherbal formulation 1. Polyherbal formulation 2, we take the ratio 1:2:1. In polyherbal formulation 3, we take the ratio 1:1:2. And in polyherbal formulation 4, we take the ratio 2:1:1. Now we do extraction. Extraction of leaves of the plant Ageratum conizoides and aglamarmelos and stem of Tinospora cordifolia were carried out through circulation method using ethanol as a solvent. Now, experimental protocol. The animal we used in the study is the male Wistar rat having weight 150 to 250 gram. The hepatotoxicity was induced by CCl4. The mechanism of CCl4 is it converts into CCl3 and CCl3O free radicals, and these radicals do membrane destruction, and LPO level in the body is increased and oxidative stress level is increased and liver fibrosis takes place. The LEAF 52 was used as a reference standard. The duration of the study was 14 days. Now we separate the animal in different groups. We are having 11 groups. Groups 1 is the control group, received vehicle 1 mg per kg per only. Group 2 is positive control group, which received carbon tetrachloride in olive oil. Group 3 is standard group, which received 52 1 ml per kg only plus carbon tetrachloride. Group 4 is test group, which received polyherbal formulation 1, which is 1:1:1 ratio of the plant extract at a dose of 250 mg per kg per only plus carbon tetrachloride for 14 days. Group 5 is test group, which received polyherbal formulation 2, which is 1:2:1 ratio of the plant extract plus carbon tetrachloride for 14 days. Group 6 is the test group, which received polyherbal formulation 3, which is ratio in 1:1:2 plus carbon tetrachloride. Group 7 is the group received polyherbal formulation 4 plus carbon tetrachloride. Group 8 is group received polyherbal formulation 1 at a dose of 500 mg per kg per only plus carbon tetrachloride for the 14 days. Group 9 is test group received polyherbal formulation 5 at a dose of 500 mg per kg only plus carbon tetrachloride. Group 10 is group received polyherbal formulation 3 at a dose of 500 mg per kg per only plus carbon tetrachloride. Group 11 is test group received polyherbal formulation 4 at a dose of 500 mg per kg per only plus carbon tetrachloride. In liver diseases, the enzyme level such as SGOT, SGPT, ALT, total bilirubin is increased and the total protein count is decreased. So we estimated the biochemical parameter such as SGOT, SGPT, ALT, total protein and bilirubin. The result of the study is that In the normal control group, the SGOT, SGPT, ALP, TPRO, and bilirubin level is normal. But in positive control group, the SGOT level is increased because we give only carbon tetrachloride in this group. We have not given the standard drug and plant extract in this group. In the standard group, the SGOT level is decreased. When we do our give our extract, the polyherbal formulation second shows a significant reduction in the SGOT level in the animals, and polyherbal formulation 1 at a dose of 500 mg per kg show the significant reduction is the SGOT level. In the SGPT, the positive control group show the significant increase in the SGPT level, while in the polyherbal formulation second at a dose of 250 mg per kg per orally shows the significant reduction in the SGPT level, while in polyherbal formulation first at a dose of 500 mg per kg shows the reduction in SGPT level. And now in the ALP level, in the positive control group, there is significant increase in ALP level, while when we give to our polyherbal formulation second at a dose of 250 mg per kg per orally, there is significant reduction in ALP level. And polyherbal formulation 1 at a dose of 500 mg per kg orally shows a significant reduction in ALP level. In the TPRO, there is significant reduction in protein level in positive control group, while in polyherbal formulation 2, when we give it, there is significant increase in the total protein level. Now we concluded that in the present study, there is elevated level of the SGOT, SGPT, ALT, and bilirubin, and reduced level of total protein has been attributed in the positive control group. While when we give to our extract in the pre-treated group with different type of the polyherbal formulation have reduced level of this enzyme compared to the carbon tetrachloride explore group. And there is the references of this research work. Thank you.
Thank you, ma'am. Any questions from the respected judges?
Yes, good afternoon. Hello, Ms. Himani.
Good afternoon.
Thank you for the presentation. This is a very interesting work. Well, I would like to just ask one question. Yeah, you mentioned here you have tested the activity of polyherbal formulation. So on what basis did you actually select all these three plant compounds, the leaves of plant and stem, right, of one plant? So on what basis did you select these three? And whether or not, I'm not sure, it's not clear from your presentation whether, you know, one or all three of it actually acts to, you know, to cause changes in the biochemical parameters. Have these been tested?
Yes, ma'am.
So my first question is, on what basis did you select these three plants? And my second question is that how do you know that all three are working synergistically and not individually?
Ma'am, according to review of literature, these three plants, Agratum conjoides, Aglamarmelis, and Tinospora cordifolia, have the potent antioxidant activity and hepatoprotective activity. So we choose this plant for polyherbal formulations.
Okay, so these are all crude extracts, right? Not selected compounds. Crude, isn't it?
Yes, ma'am.
Okay. All right. So how do you know, or how can you be sure that they work synergistically and not individually?
Sorry, ma'am, I'm unable to listen.
How do you know that they act synergistically and not individually?
Ma'am, when we give this plant extract separately, there is a low reduction in these levels. When we give 3 extract combinatorily, then there is significant reduction in these levels in groups.
Okay. But is it shown here in your table?
No, ma'am.
Yeah, so I'm not sure whether, yeah, so that is something that I wanted confirmation. Yeah, so that's all from me. Thank you very much.
Thank you, ma'am.
If the other judge has anything, they can ask.
Thank you, ma'am. Any more questions from the respected judges? Ma'am, can we move to the next presenter with your permission?
Yes, sure.
Thank you, ma'am. There is a small announcement for the participants of Hall B and Hall C. Please join the breakout rooms. I request, please join the breakout rooms. Thank you. I'd like to invite the next presenter, Samrat Bose, sir, on oral presentation on assessment of toxicity and safety margins of phenylalanine and metabolic through in vivo and vitro models of zebrafish.
So am I audible?
Yes, sir, you are audible.
Whether my screen is full screen or I need to make it full screen?
Yeah, sir, please make it on the slideshow. It would be better.
Just a minute.
Yeah.
Good morning to all the respected participants and judges. So myself, Samrat Bose from JIS University, Kolkata. So today my topic of presentation is assessment of toxicity and safety margin of phenylalanine and its metabolites through in vitro model of zebrafish genuine. So before I start to talk about my work, so the introductory part, I can say that as per the statement of WHO, that most of the developing nations, the main resource of healthcare system is traditional medicine practice along with the food supplement. So when we are talking about the food supplement, sorry.
I'm so sorry for inconvenience, sir. I request other participants to mute yourself, please.
Yeah. So when we are talking about the food supplements, it's not about the vitamins or minerals, but amino acids having another play or another important role in this part. So it is also reported in a reputed journal that lack of clinical safety information on amino acid use in general populations. And considering the country like China, Indonesia, Thailand, they do not currently have a regulatory mechanism governing the uses of amino acids, and thus do not allow such uses in general. It clearly denotes the safety concern of amino acids. Countries in Asia like Japan, Korea, Singapore, and Latin America do not directly supervise amino acids in the dietary supplements. That is the alternative called the food supplements. Now, considering our my main objective related with the essential amino acid, that is the phenylalanine, it is already reported the metabolism may lead to several clinical manifestations, including growth failure, seizures, and intellectual impairment. It is also reported that it may cause the acute respiratory distress syndrome associated with the high level of plasma phenylalanine. Now, if we concentrate on the part of its metabolism, the phenylalanine can be metabolites into 3 different ways. One is tyrosine pathway, another is phenylethylamine metabolism, and another is phenylpyruvate metabolism. Where the main cause of the conversion of phenylalanine into tyrosine is where the responsible thing is the phenylalanine hydroxylase, that enzyme. If that availability of the enzyme is not there, automatically the availability of phenylalanine will be higher, and parallelly the phenylpyruvate availability will be much higher. But there is no such confirmation or any published literature where we can identify that exact which metabolite is responsible for such kind of outcome. So the main objective of this study is to identify the role of phenylalanine and its metabolites in embryo toxicity, development toxicity, and teratogenicity study. So as I have already mentioned, for this study, I have selected zebrafish because of its certain advantages like 70% gene similarity considering the protein coding gene and 84% disease causing gene similarity is there. It also assures the compliance with the 3R principles like replacement, reductions, and refinement. We can also use this model for genocardiotoxicity determination because the electrophysiology of zebrafish heart and human heart is almost same, although zebrafish heart is just a two-chambered heart. And also we can use this model for off-target effect determination. So now plan of work is the collection of the dengue radio. We have to breed it because initial study has been done on the embryo. So we need to observe the developmental stages, procurement of drug. In this study, I have considered phenylalanine and one of its metabolites, that is phenyl pyruvate. I have performed LCVP study as per OECD guideline 203, mortality morbidity study as per the OECD 236, developmental toxicity study as per the OECD guideline, and also the biochemical estimations I have done, SGPT, SGOT, and for its molecular assessment, I do consider the histopathological assessment of hepatic cells. So why what are the endpoint of 236 test model? The endpoints are coagulation of fertilized egg, lack of somatic formations, lack of detachment of the tail bud from the yolk sac, and lack of heartbeat. These are the endpoint. Now I have collected, separated the male and female zebrafish in my labs, and these are the materials which are used for the breeding purpose. And for the breeding purpose, I need to maintain the temperature 26 plus minus 1 degree Celsius. For that purpose, I have used one thermostat, and also E36 solution. E3 solution is very much required for the collection and for the measurement of further studies, because we need to keep the eggs in E3 solutions. Another very important parameter is what are the foods. I have used the commercial aquarium fish food, which is approved as per the OECD guideline, and to feed the embryos, I have used the brine shrimp. Now, these are the real-time picture which has been taken, because as my objective is to identify the deformities at different stages of the embryo. First of all, I need to understand what are the different stages can be observed. So these are the real-time image where you can see easily at the different time interval how from the eggs the entire embryo has been formed. These are the period where 0.75 hours, where cleavage period, blastular period, as you can see, the gastular phase, the segmentation occurred, and finally you can see the hatching and the embryos. Now, as for the LCVP study, for the phenylalanine on the embryo, I have selected 6 different concentration of phenylalanine, and I have observed the development from 24 hours to 120 hours. Although as per the protocol, I can end it at the 96 hours, but I have performed it up to 120 hours, and these are the results. This is very interesting that at 24 hours, the LD50 value for the phenylalanine, which is around 2000 microgram, and gradually it is decreased, and at 120 hours, it is 92, which is also shown in this graph. The same thing, in considering the mortality study, which is also interesting, that at their initial 2 lower concentration at 50 microgram and 60 microgram, where the mortality rate is very less, which is around 40% max to max, but considering the higher concentration, that is 250 and 300 microgram per ml, where the mortality rate has reached around 100% or nearest to 100% within 96 hours, as you can see in this graph. Similarly, I have performed the same LCP value of the metabolites, that is phenyl pyruvate, where the outcome, as you can see, at 24 hours, the amount is 224 microgram per ml, which is much lesser than the phenylalanine. As we are studying on the phenyl pyruvate, that is the metabolites, and it has gone down up to 79 microgram per ml at 120 hours, which is also shown in this graph. So from these 2 studies up to this, we can say that yes, phenylalanine is much safer than the phenyl pyruvate up to this, not yet. Now, considering the mortality study also, the initial 2 concentration have shown 20% mortality rate and 40%, but at the highest concentration, which is very significant, even at 72 hours, the mortality rate has reached 100% for 350 microgram per ml, also for 250 microgram per ml. It is almost reached 100%, as you can see from this graph. Now, to access the developmental toxicity study, I have considered 3 different concentrations of phenylalanine, which is much higher than the phenyl pyruvate, as I got the information from LCVP study that phenylalanine is much safer at its highest concentration, but phenyl pyruvate is lower. And I have considered 4 different parameters, although I can consider 6, 8 different parameters, but I have considered 4 different parameters: 1 total body length, head width, and cubital cylinder. I have concentrated more on the head length and head width because phenylalanine is another. It has another application, that it has a significant role in neuronal development also. That's why I choose these 2 points, head length and head width, and give more importance to that. For this purpose, I have used image software and few parameters. If you use image software, few parameters has to be maintained. So these are the 4 basic parameters. I don't want to explain all those things, but these parameters are very important. Those who want to use the image software. And these are the scale set. You need to use the scale set first into 4 different stages. Then only you can use. So these are the steps which you must follow during the assessment through this image software. So in this way, I have performed the assessment, that means the head length, head width, caudal fin length, and total body length by applying this software. I don't want to explain all those things. These are the results. So let's see the statistical interpretation. So considering the total body length, I have used 3 different concentration of phenylalanine 0.5, 0.75, and 1 milligram, and sodium phenyl pyruvate 0.25, 0.4, and 6 milligram per ml concentration. And it is significantly observed that in case of phenylalanine, there is no significant change in total body length, but the body length has been drastically reduced in case of sodium phenyl pyruvate. Although by the application of phenylalanine, it is slightly higher, but there is no significant in comparison to the control. Considering the head length, there is a correlation in between the phenylalanine at its highest concentration, which is at 1 milligram per ml, and the highest concentration of sodium phenyl pyruvate. But if you closely observe the graph, in case of the application of phenylalanine, although the head length has been increased, but in case of sodium phenyl pyruvate, it is significantly decreased and in graduated manner. But the highest concentration is showing the significant one. Now let's talk about the head width. In case of head width also, there is a significant difference in case of highest concentration of phenylalanine, but again, the head width has been decreased significantly in different concentration. But when I perform the ANOVA, as per the statistical result, there is no significant difference has been shown. Only in case of phenylalanine is shown. In case of caudal fin length, where the highest concentration of phenylalanine as well as highest concentration of sodium phenyl pyruvate has shown significant difference, divided by 3 star. Now I have performed the biochemical estimation considering SGPT, SGOT by applying the drug at 3 different concentration, 10 milligram, 20 milligram, 40 milligram per kg per intraperitoneal route, where I have observed that in case of phenylalanine, only at its highest concentration, that is 40 milligram per kg body weight, has shown the significant difference. But in case of sodium phenyl pyruvate, the results are significant for all 3 concentrations. That is, SGOT level is higher in case of application of sodium phenyl pyruvate, which is actually the metabolites. The same thing has been observed in case of SGPT, where there is no significant difference in case of a different concentration of phenylalanine. But in case of sodium phenyl pyruvate, the differences are shown very clearly. Now to check the molecular changes, is there any molecular changes at hepatocytes? I have performed the histopathological study with the same concentration, considering 10, 20, 40 milligram body weight. Initially, I have started with the phenylalanine, and I have chosen 6 different parameters histopathological assessment. One is sinusoidal dilatation, cytolysis, karyolysis, cell-to-cell addition decrement, vascularization, and degenerated hepatocytes. After performing the experiment and observing the histopathological slide, I can conclude that only in case of the sinusoidal dilatation, slightly difference has been observed, and at its highest concentration, only cell-to-cell addition decrement has been observed in case of phenylalanine. So there is no major significant change has been observed under the histopathological study under 3 different concentrations. Only at highest concentration of phenylalanine, 2 minor changes observed. But when I check the same thing on the metabolite, that is sodium phenyl pyruvate, considering all 6 parameters, significant changes has been observed in case of sinusoidal dilatations, karyolysis, cell-to-cell addition decrement, vacuolization, and degenerated hepatocytes. And mostly at its highest concentration, also gradually it has increased from its lowest to highest concentration, that is 10 milligram per kg to up to the 40 milligram per kg body, which is clearly shown in this figure. So already I have discussed the results. So now I can conclude by saying that phenylalanine is safe at 10, 20, 40 milligram. Only negligible changes observed in developmental, biochemical, and histopathological study. But phenyl pyruvate is not safe as it causes different developmental abnormalities, biochemical changes in SGPT, SGOT, and also showing other hepatocellular abnormalities in histopathological study, mostly at 20 and 40 milligram per kg. Therefore, consumptions of amino acid containing dietary supplement, individual amino acids or in combination may cause potential risk to the subject. So the expected outcome may enlighten that the development, this study may useful for the development strategy of consumption of amino acid containing dietary supplements, individual amino acids or in combination or of its where potential risks of the subject will get reduced. So up to this, these are the reference. Thank you very much. So if there is any question.
Thank you so much, sir. Any questions from the respected judges? Any questions from the respected judges?
Yes, good morning, Samrat.
Good morning, sir.
And good morning, organizers. Sorry, I just joined. Not able to follow your full presentation. But so there is no question from my side. Okay, so if I heard the question presentation, I can ask. So I let the other judges can ask the question. Not from okay.
So can we move to the next
Yes,
presentation?
yes.
Yeah. Thank you, sir. Thank you, Samrat, sir. I would like to invite Jamwant Kumar, sir, for presentation, e-poster presentation on neuropharmacological activity of ethalonic extract of Elanthothophis scaber lin leaves.
Yes, ma'am. Good morning, ma'am.
Good morning, sir.
Ma'am, my topic, ma'am. My
Yes,
name
sir.
is Jamant Kumar. My name is Jamant Kumar from Varanasi, CET College of Pharmacy. My topic name is a neuropharmacological activity of ethanolic extract of Elephantopas scaber leaf. Okay, introduction. The lectotype species of Elephantopas genus, example में Elephantopas scaber leaf, family Asteraceae. It is common wild seed that for undergrowth in shady place, in rather coarse, in rigid, erect and hairy hair, 30 to 60 centimeter height. Elephantopas scaber has been widely used in traditional medicine for various diseases like fungal disease. It is used in anti-inflammatory, antipyretic, diuretic, anti-coagulant, antibiotic, and emollient and tonic. In India, it is used in the case of cardiovascular disease, bronchitis, smallpox. Root powder mixed with boiling water is given internally to cure burning sensation and gonorrhea. Root is used in heart and liver trouble, and hot water extract is root given the filaria. Ma'am, material method. Material I found in Elephantopas scaber leaf, Varanasi, Uttar Pradesh, ethanol and pentylintrazole and diazepam. ओके अब क्या मैम जो मेथड में मेनली क्या है जो आई हैव डन मैक्सिमल इलेक्ट्रोशॉक सीजर एंड पेंटिलिन ट्राजोल इंड्यूसेस सीजर और नेक्स्ट डिजीज है मैम इलेवेटेड नेक्स्ट मॉडल है मैम elevated plus means and open field model for anxiety. Okay ma'am. So I have result ma'am. We have prepared three test group ma'am. First 100 mg per kg body weight drug in rat model and 200 mg per kg and 300 mg per kg. I got result, ma'am. 100 mg is not satisfied giving result, but 2000 mg. I gave satisfactory result, but 300 mg also good. Yeah, good result, ma'am. Yeah, ma'am.
Yes, sir.
Yeah, ma'am. Complete.
So have you done?
Yeah, ma'am.
Okay. Any questions from the respected judges? I request Shubhankini Patil, ma'am, please mute yourself.
So, have you got this ethical committee for the carry out this study, Jamal?
What, sir?
Have you taken the ethical committee permission to carry out this study?
Yes.
So how you carried out this neuropharmacological activity? With the permission of the ethical committee clearance, is there or not?
Yes, sir. Yes, sir.
Why can't you
From my
mention
college,
that
sir.
ethical committee clearance number in the materials and methods itself? So before carrying out any animal experiment, you need to submit the ethical committee clearance, right? So wherever you have been presenting, you need to show the number of your
I forget out, sir. I forget out. Note down, sir.
So please keep this in mind always. Okay. So
Yes.
so then only your study will be ethical approved. Okay.
Yes.
Otherwise your study will not be considered. Okay. So
Yes.
you have taken how many extracts? Only ethanolic extracts. Ethanol extract alone you have been seeing.
Yes.
The poster should be broad now, so I am not able to see it full size. Yes.
Vishal Srivastav, sir, please mute yourself.
Okay. So the place you have collected from Varanasi, Uttar Pradesh. Okay.
Yes, sir.
So have you checked the scientific already the activity done? On what basis you selected this plant?
Because I have found in very references, sir.
Can you give me some examples how you gone to your literature review? Some short brief you tell me. What are the books you have referred?
S.K. Kulkarni, sir. KDT.
S.K. Kulkarni, the plant is there in that?
No, sir. Plant literature, sir.
That's why I'm asking. Can you give me some examples? See, we all in all states we have some folklore climb, right? So that the activity cannot be proven scientifically. So some folklore climb will be there. On what basis you selected this plant?
Indian Ayurvedic pharmacy, sir.
Okay.
Pharmacopoeia. Indian Ayurvedic pharmacopoeia.
Some one book is there. How so authentication? How you have done this authentication? Who done the authentication?
Sir.
Huh?
What, sir?
Who done this authentication? This plant identification. Some confirmation we have to get from botanist, right?
Yes, sir.
So they have to authenticate. Yes, this is the plant.
Yes.
Elephant,
yes, sir.
Elephantophus scaber. Okay, this is the plant of.
Yes.
So we never seen the Elephantophus scaber previously. So you were also been never understood. So you may heard about that, and you have to. But we are not in position to describe whether this is the Elephantophus scaber plant or not. Okay, so how you identified this plant? Some verification has to be done by some botanist, right? So these procedures all you know or not previously?
From BHU, sir.
You identified a certificate is
yes,
there
sir.
from them?
Certificate, sir, I have.
You need to get a signature and you have to keep it back. So this is your
Yeah.
PhD work or any other work?
No, sir, M-Pharm.
M-Pharm work. Yeah.
Yeah. Yes.
Anyhow, all these are the basic procedures before carrying out this plant studies.
Yes.
Okay. So
Yes.
these are the
Yes.
okay. And
Yes.
so ethanolic extract. What is the all these
Thank you, sir.
elevated plus mass everything anxiolytic effect only have been all neurological effects, right?
Yes.
Electroshock seizure, induced
Okay.
convulsions. Okay, so how they are given 25 rats, 5 groups,
What's it?
and ethical
5
committee
groups.
approval is there. They are
Yes,
given
sir.
5 rats.
Yes.
Have you sacrificed any animal?
No, sir.
No. Okay. So what about the results? Only
Sir, I will mention, sir, in
given
the
some graphs, but we are not been that much clear in that. Okay.
sorry, sir. I will mention, sir.
Okay. Anyhow, good. Okay.
Yes.
So
Thank you,
please
sir.
keep all these points in mind. Okay, before going
Yes.
for any presentation, we need to mention it that. Otherwise,
Yes,
materials
sir.
methods in the beginning
I will
itself, 2
mention.
lines you need to add. Okay. So next
Yes.
presentation we will. Okay. Thank you.
Yes, I will mention, sir. Thank you,
Okay.
sir.
Thank you.
Welcome.
Now it's open to other judges.
Yes, sir. Any more questions from the respected judges? Sir, can we move to the next one with your permission?
Yes, yes, yes.
Thank you, sir. I'd like to invite Ratna Priya ma'am from Sardar Bhagwan Singh University, India, e-poster presentation on modulary effect of wheat sprout extract and melatonin and myocardial reperfusion injury in rats.
Yes, ma'am.
Yeah, I request Jamwant sir, please stop your screen. Yeah, thank you.
Okay, ma'am.
Questions are over.
Yes, ma'am. Is my screen visible?
Once again.
Yes, yes, ma'am, it is visible.
How many presentations are over, ma'am? This is Rakibe.
Yes, sir. Sorry, sir.
How many presentations are over, ma'am? 3, yeah? 3, yeah.
Yes, so this is fourth one.
This is the fourth one. Yes.
Yeah, it's the fourth one.
Okay.
Yeah.
Okay, so good morning, everyone. My name is Ratna Priya and I am from Sardar Bhagwan Singh University, India. So the topic for my presentation is modulatory effect of wheat sprout extract and melatonin in myocardial reperfusion injury in rats. So first of all, what is myocardial reperfusion? So myocardial reperfusion is the restoration of coronary blood flow to the ischemic areas. So why does it cause injury? So injury can occur because of post-ischemic reperfusion. So here there is removal of antioxidant. So during ischemia, there is a high amount of antioxidant that gather in the area of ischemic tissue. So when the blood flow is restored, that antioxidant washes out, so causing an oxidative stress and cellular damage. Further, some other things can also happen, like increase in intracellular calcium, because the sarcoplasmic reticulum can get damaged during the ischemia. So because of that, the calcium are unable to get inside the sarcoplasmic reticulum, and the level of intracellular calcium is increased. Further, inflammation can also be a cause for this injury, and neutrophil endothelial interaction can occur. Further, free iron mobilization can be seen that lead to cardiac myocyte damage. So now I am using wheat sprout extract. So why wheat sprout extract? It is because it is rich in carotenoid and phenolic compound. Further, there is high amount of bioflavonoid in it. Then melatonin is also used as melatonin act through the MT2 receptor activation, and it increases the myocardial function, so the energy will be increased further. There is also found an evidence that it arrests the cellular and molecular damage. So that's why we are using melatonin here. So further, coming to the experimental design, animals were divided into 6 groups, and 6 animals were taken per group, and the treatment was followed for the 30 days. And after treatment, induction of myocardial reperfusion injury was performed. So the treatment group, in treatment group, normal control, positive control, wheat sprout extract at a dose of 10 mg per kg per oral was given by in distilled water. Then melatonin at a dose of 10 mg per kg per oral was given as a suspension. Further, the combination of wheat sprout extract and 10 mg per kg per oral and melatonin again 10 mg per kg per oral was given in a combination as suspension form. And another combination is also given in which the wheat sprout extract and melatonin 15 mg per kg per oral was used for the both of extract in combination form as a suspension. So these treatments are followed for 30 days. Then after the treatment, induction of MRI, that is the myocardial reperfusion injury, was performed. So how does it perform? So for that, we administered anesthesia, that is pentobarbital at a dose of 60 mg per kg IP, and along with the anesthesia we provide atropine. So why atropine? Atropine is given so it steady the heart rate, and it also decreases the tracheobronchial secretion. So further, the chest was open and the pericardium was also open as well. The LAD artery, that is the left anterior descending artery, was ligated with the help of a suture for 15 minutes to induce ischemia. Then, after 15 minutes of the cardiac reperfusion was allowed by releasing the ligation. So, after this, the sutures are used to close the skin and bind the ribs. So, after the induction of MRI, after 3 days, further different parameters were performed. Okay, so for that parameters to be estimated, they are like biochemical parameters and oxidative parameters. So in biochemical parameters, we observe calcium level, SGOT, LDH, troponin 1 and troponin T. And in oxidative parameters, LPO, SOD, GSH and catalase activity was estimated. So further coming to the result portion, so the calcium in control group, there is an increased level of calcium as the intracellular calcium is increased in this condition. So wheat sprout extract and melatonin at a high dose level, that is 15 mg per kg both, shows the most significant result in decreasing the calcium level, the serum calcium level. Further, the LDH level is also need to be decreased as LDH is released when the tissue is injured. So for that, the wheat sprout extract and melatonin shows a very great result, but the wheat sprout extract at a dose of 10 mg per kg per oral has the most significant effect in this. Now, SGOT. So the SGOT level is decreased with the help of wheat sprout extract 15 mg per kg and its combination with melatonin 15 mg per kg per oral, and it shows the most significant effect here. But wheat sprout extract at a dose of 10 mg per kg also has some effect on it. Now, coming to troponin 1 and troponin T. So troponin 1 and troponin T, these are the biomarkers for cardiac injury. So it is released when there is cardiac injury. But here, in this case, we can see in control group they are present, but the traces are absent in every other group. So that means that there is no cardiac injury, or there is a very less amount of cardiac injury, because of which the level of troponin 1 and troponin T is very less. Now coming to the oxidative parameters. So for oxidative parameters, SOD, catalase, LPO and reduced glutathione, that is GSH, is estimated. So here also the wheat sprout extract at 15 mg per kg per oral and melatonin at 15 mg per kg per oral so shows a very significant result by increasing the SOD level and catalase as well. Further, it significantly decreases the level of LPO, and in reduced glutathione, the melatonin alone at 10 mg per kg shows the best result in increasing the level of GSH. So these are the graphical representation of the result mentioned above. Now coming to the discussion portion, wheat sprout extract and melatonin maintain the level of GSH, SOD, catalase, as well as it decreases the level of LPO. Treatment with wheat sprout extract along with melatonin significantly decreases the LPO as it shows the better results. Then further, the troponin 1, troponin T and SGOT level is also decreased. So rather than carotenoids, wheat sprout extract also has a conjugate dienes, and because of that it has a very high antioxidant effect. So because of this, the following result was obtained. So these are the references. Thank you.
Thank you, ma'am. Any questions from the respected
Yes,
judges?
yes, ma'am. Very good presentation.
Thank you, sir.
The Ratnapriya, huh? And
Yes, sir.
uh, have you got the ethical committee clearance to carry out this study, ma'am?
Yes, sir. Sir, actually, I am the presenter here. There are other people who are involved in
Okay,
the study.
okay.
Yes,
It's
sir.
a group project, right? Okay.
Yes,
So
sir.
you are a faculty or a student?
Sir, I am a student of M.Pharm Pharmacology and I am in my
Okay,
second year right now. Yes, sir.
okay, okay. So have
Yes.
you got ethical committee clearance, right? That's why I'm asking. You need to mention some ethical committee number, approval number
Oh,
in your presentation. So wherever
okay,
you are
okay,
doing
sir.
your presentation or publication, we need to
Yes,
mention
sir.
because nowadays you know very well that animal ethics committee is more serious than human ethics committee.
Yes,
Okay,
sir.
so
Right.
we need,
Okay, sir.
yeah,
I'll
okay,
take care of
because
it.
anyhow with the intervention studies we are doing compulsorily, we need to submit the ethics committee. And
Yes, sir.
regarding the wheat sprout extract, whether you have
Yes, sir.
any evidence how you chosen the wheat sprout extract, on
Sir, there
what
are
basis?
certain there are certain research article and review also based on that there is high content of phenolic compound, and as we know phenolic compound is able, it has a high antioxidant effect. So here the main problem is oxidative stress, so because of that we thought that it will be able to decrease the oxidative stress by its antioxidant effect. That's why we use wheat sprout extract here, sir.
So
Yes,
your
sir.
which concentration has been very effective,
So
proven to
here
be
the
very
wheat
effective.
sprout extract, when we give it with melatonin, at a dose of 15 mg per kg for both,
Hmm.
they shows the most significant effect.
Okay, so
Yes,
troponin,
sir.
yeah, all cardiac markers has been very effective. 1, 2, 3, and let's see. Okay,
Yes, sir.
except carotenoids, which shows a higher number of consumer factors. That is protective potential. Okay, so from
Yes,
here, what is your plan? You have proven that
yes,
it
sir.
is a very effective cardioprotective potential. So from now on, what is your direction regarding this study?
Sir, basically, sir, this myocardial reperfusion injury, it can be very dangerous. So that's why we use this thing, and we can see here that it help in reducing the level of troponin as well. The troponin, they are present in control group, but except that there is the no traces of troponin seen. So it may be responsible for healing, not
Uh,
healing exactly, but to prevent the injury. These, uh,
okay,
yes, yes,
okay. So I am asking, you already proven that, right? It's a cardioprotective potential is there. Okay, so from now, what is your plan next? You maybe need some trials, right? Preclinical studies you need to carry out, and you have to
yes, sir.
prove yourself. Okay, so this is my suggestion because you
Yes, sir.
have been done a wonderful work and it needs to be carried over to the next level. So
Yes.
don't drop here itself. Okay,
Okay.
it is
Yes,
a
sir.
very good study. Thank.
Thank you so much, sir.
Because it's a very economic wise, it's very cheap. The wheat sprout, we have been taking that means we
Yes,
don't
sir.
have much uh, amount involved in this. Okay, so it may be very useful and very effective. Thank you.
Yes, sir. Okay, sir.
All
Thank
the
you,
best.
sir. Thank you
Go
so
to
much, sir.
the other judges.
Yeah, thank you, Ms. Ratnapriya. That's it's really a very interesting study, right? You're using wheat sprout extract with melatonin. Well,
Yes.
since you have got very good results, right? You've shown that it maintains, certain levels of the GSH, SOD, and CAT. So what do you think, how can you explain the synergism between these wheat sprout extract with melatonin?
Okay, ma'am. So according to the results, I'll say that when wheat sprout extract and melatonin, when they are given alone, they show some result. Like they are also lowering calcium level. They are also showing a result. But when they are given in combination at a particular dose, like when we give it in 10 mg per kg, there is a group where I am giving it in 10 mg per kg for both. They are not showing that much good result in compared to the group where I am providing it at 15 mg per kg for both. So ma'am, according to this, I think at a particular dose level, they work together to lower the certain like SOD level, they increase the SOD level, they work in a synergistic manner at a particular dose.
Okay, so you are saying that it's on a dose-dependent manner, right? But I think, coming back to following Dr. Karthikeyan's question also, the future plan, I think you would need to, if you've got very good results like this, then you would probably need to have some kind of explanation and maybe some research to explain or discover the mechanisms of action for both synergistically. What do they actually do, right?
Yes, ma'am.
Yeah, in maybe looking at protein levels or gene level expressions, something like that, right? Have you done it?
No, ma'am, I haven't done it.
This
These
is
are the
a preliminary data, is it? Okay.
Yes.
All right, so that's good. Your preliminary data looks good, and I wish you all the best. Thank you very much.
Thank you so much, ma'am.
Okay, we can move on to the next presenter.
Yeah,
There's no other questions.
Sure, ma'am. Thank you, ma'am. Thank you, sir. I'd like to invite Reshma ma'am from Deccan School of Pharmacy, India. Oral presentation on right pleural effusion with right mass lesion.
Okay, ma'am.
Yeah. Please share your screen, ma'am.
Yes, ma'am. Just a minute.
Yeah.
Is my screen is available, ma'am?
Yeah, it is visible.
Okay. Okay, very good morning, respected teachers and my dear friends. My name is Reshma, pursuing M.Pharm second year from Deccan School of Pharmacy, which is affiliated to Osmania University. So today I'm going to give a oral presentation on right pleural effusion with right mass lesion. So the right pleural effusion with right mass lesion, we have to know what is right pleural effusion first of all. So it is nothing but a buildup of excess fluid between the layers of pleura outside the lungs. So mass lesion in right lung is nothing but the lesions larger than, if it is larger than the 3 centimeter, then it is considered as the masses and are treated as cancerous until it is proven. And then there are two types, that is a transudative and the exudative. In transudative, it forms when there will be a leaking across the normal pleura. It should be drained off if it is very large. The common cause for this, it is a congestive heart failure. And next one is the exudative. This forms from the extra liquid or protein, blood, inflammatory cells, bacteria, and that leaks across the damaged blood vessels. It should be drained off depending on the size and the inflammation where it is. The cause for this type is pneumonia and lung cancer. The causes for this, it is a wide range of things. Some of them are leaking from other organs, usually happens if some have the congestive heart failure. And when in congestive heart failure, there will be a it doesn't have a pump blood to the body properly, so it occurs. But if it also come from the liver or kidney disease, and usually lung cancer is the problem, then the other cancers which have spread to the lung or pleura can cause it. And these are the diagrams where we can see there are the two layers, that is the visceral pleura and the parietal pleura. The visceral pleura is the blue one, which is the inside the layer of the lung, and the parietal pleura is the outer side layer of the lung, and there is a space between the two layers known as the pleural space. And it is also explaining the transudative pleural effusion and exudative. In transudative, it occurs when the hydrostatic pressure or lower plasma oncotic pressure is increased. Oncotic pressure is in the sense if the part of the total osmotic pressure of the plasma that is due to the impermeant of the proteins. Example, cirrhosis, PE is nothing but the pulmonary embolism, and CHF, that is the congestive heart failure. And here the pleural effusion, as we all know, if there will be the accumulation of fluid within the pleural space, it is known as the pleural effusion. And exudative is it occurs when the inflammation and increased capillary permeability. The examples for this exudative is the pneumonia, cancer, TB, viral infection, autoimmune diseases. And here we can see the diagram, low in protein and LDH. LDH is nothing but it is a enzyme. LDH is full form lactate dehydrogenase. It is a maker of inflammation or cellular injury. If the LDH levels are greater than 3 times, then the normal limit often indicates as pleural infection. Low protein causes pleural infusion. High protein can makes it to be a malignant, then a transudative effusion. So the infections, some of them include the that lead to pleural effusion are pneumonia or tuberculosis, and the autoimmune conditions are lupus or rheumatoid arthritis are some of the diseases. Pulmonary embolism is nothing but if there will be a blockage in an artery in one of the lungs, and it can lead to a pleural effusion. Symptoms may include the shortness of the breath, chest pain, fever, cough. The diagnosis is doctor will talk about the symptoms first of all and gives a physical exam, that is a tap on chest and listen with the stethoscope to confirm if the person have a pleural effusion. Some of the tests are chest x-ray, pleural effusion appear, chest x-ray gives the results as pleural effusion appear white on x-rays, while A space looks like a black. And we can also go for the CT scan. It is a computer construct images of the entire chest, inside and outside. And also ultrasound, doctors may also go for it. In ultrasound, the procedure is known as the thoracentesis. They'll take a little bit of the fluid to test. They insert a needle and tube called a catheter between the ribs into the pleural space. The standard treatment for this is antibiotics for pneumonia are given, and or diuretics for the congestive heart failure. Large infected or inflamed pleural effusions are often need to drained off to feel better and prevent more problems. The procedure for treating pleural effusions include thoracentesis, tube thoracostomy, and pleural drain by urodiasis. In thoracentesis, if the fusion is large, doctor may ask to go for testing just to ease your symptoms. And tube thoracostomy is a chest tube where a small cut in the chest will be done, and they put a plastic tube into the pleural space for several days. In pleural drain, and if the pleural effusions keep coming back, then the doctor has to put the long-term catheter through skin into the pleural space. Then the pleural effusion can drain at home. Next to the pyurodiasis, doctor injects an irritating substance such as like a talc or doxycycline through a test tube into the pleural space. This pleurodiosis can prevent pleural effusions coming back. The SOAP format, by taking a lady survey, the lady is of 45 years old, female, was admitted in the pulmonology department with the complaints of cough, SOB, fever, grade 4, and since 4 days. And she has a past history of tuberculosis, which was treated 6 months back. Present history is DM, that is diabetes mellitus, and the patient was apparently normal all the night of 5 days back. she developed a protective curve, SOB, and fever. Provisional diagnosis was asthma exacerbation and acute hypoxemic respiratory failure. The objective data was the chest X-ray, which was found as pleural effusion was found. Bronchoscopy report, right lower lobe bronchus narrowing, and bacteriology report was no bacterial growth after 48 hours aerobic incubation. And the Graham stain report was no pus cell seen. No microorganisms were seen. Microbiology report was fungal element, negative for the fungal element. Bacteriology report, acid stain, negative for acid fast bacilli. And the HIV 1 or 2 non-reactive. HBS AG was negative, and hepatitis C was non-reactive, and RBS is the random blood sugar for the lady was 182 mg per dl was increased. Final diagnosis was done right pleural effusion with right mass lesion. So these are the treatment given for the 12 days. For day one, uh, in day one, the patient was complaining about the grade 4 fever, cough, and OE conscious coherent. Temperature was 99 degrees Fahrenheit. BP was 110 for 70. Pulse rate was 84. RR was 20. And these are the symptoms from day 1 to day 12. And these are in a tabular form it is given. In temperature was 99 or 98. BP was maintaining in this way, sometimes 110 to 80, 130, 120 by 70. And the pulse rate was 90, 84. It was up and down. And GRBCS is, this is the values for GRBCS and RR per minute, and SpO2 was this. These are the values. Then the treatment was given for that lady was injection forms, injections or tablets or nebulization. Drug was Taxim, generic name cefotaxime, category antibiotic, dose 1 gram, route was IV, frequency BD. Duration for day 1 and day 13. These are the tablets and nebulization and injection which was given to the lady from day 1 to day 13. For the assessment, in assessment, the problems were, first problem was shortness of breath and cough. For that, medication was given, levosalbutamol and ipratropium, and budesonide levosalbutamol. For problem 2 was the fever. For the medication given for the fever is eterocoxib and paracetamol. Problem 3 was DM, diabetes mellitus. The medication given for this is glibenclamide and metformin. Problem 4 was pleuritic pain and headache. The medication given for this is paracetamol and Ramadol. Problem 5 was nausea and vomiting. The medication was given Zoffer 4 mg. Problem 6 was abdominal discomfort, and the medication given is pantoprazole. Problem 7 was mass lesion and pleural effusion. Medication was given cefotaxime and hydrocortisone. And these are the treatment which is given for the 5 days. And the patient counseling was done regarding disease. The pleural effusion is filled up of the between the space between 2 layers of the pleura. Pleura is a thin space of tissue within the layers. One layer rests directly on the lungs, the other rest on the chest. There is normally a small amount of fluid between these layers. Fluid helps lungs move easily when you breathe. Regarding medication, it should be taken on time. Do not skip any dose if you feel better, and stop this if you feel itchy or rashes. Tablet omeprazole D, take it 1 hour before meal. Inhaler, rinse before taking, after taking or before taking. We have to rinse the mouth with water after each use to prevent infection in mouth. Carry your inhaler wherever you go. These are the patient count. This was the given to the lady this is known as the patient counseling. The lifestyle modifications, pressure to decrease pain. We have she should take the pillow when she cough or take a deep breath. Do not allow others to smoke around her. If they smoke, that risks her lungs infection such as pneumonia. Smoking also makes harder for you to get better after you have a lung problem. Deep breathing and coughing will decrease your risk of a lung infection. So she should eat healthy and balanced diet with plenty of vegetables, fruits, and lean proteins. The limit sugar is fat and maintain healthy weight. Healthy eating is important during and after the treatment. And she should manage the stress and aim for 7 to 8 hours of sleep a night. Get it in the habit of washing hands frequently to avoid the viruses or bacteria. If she feels like a cold or viruses, she should get the plenty of rest and seek medical attention. If the chest pain, difficulty breathing, or fever, or if any symptoms are worsen, she should get the medical attention. This was the counseling and lifestyle of the modification was given to the patient. Thank you.
Thank you, ma'am. Any questions from the respected judges?
Go to the soap notes, ma.
Sir?
Please go to the soap notes, please, ma. Soap notes, subjective.
Yes, sir. Yes, sir.
45-year-old female, right? With the complaint of cough, grapefruit. Go to the treatment chart.
Sir?
Treatment chart, ma. Treatment planning.
This is a treatment.
Why 2, 3 steroids? First we have gone for injection. Then again injection.
Sir?
Steroids, so much of steroids being included. Is it really necessary for this patient?
According to the patient's health, it is given, sir.
That's why, ma. Here, sumac hydrocortisone, IV, they given. Here also IV on day 1. From day 2, they changed that.
Yes.
Again, 100 mg, this is 100 gram.
Yes, sir.
Tramadol, what for they given? Tramadol.
Sir?
Tramadol and paracetamol. For what purpose they given? Fever. For fever they administered.
Yes, sir.
What is the necessity of giving tramadol, ma?
Tramadol.
The last drug.
Where it is, sir.
That's it. Coming the opioid analysis. Is there any pain or anything the patient suffer from?
Where it is, sir, Tramadol.
Ultrac, ma, last drug.
Okay, Tramadol, yes, sir.
Yeah. Why they are given tramadol here?
If there will be a pain in the chest, it is given, sir.
Chest pain. Okay. So you will give the tramadol directly. Tramadol coming under you just given opioid analgesic.
Yes, sir.
What category it comes under opioid?
Opioid.
What analgesic?
NSAID.
NSAID.
Yes.
Narcotic analgesic, ma. What it will happen, ma? Simply See, nowadays more prescription is there using this tramadol. Okay.
Okay.
You are a PharmD graduate, a PharmD student, or a pharmacy graduate?
Sir?
You are a PharmD student,
No, sir. M-Pharm student.
M.Pharm?
M-Pharm second year, sir.
Which specialization?
Pharmaceutical analysis. Actually, I did this subject with the help of my friend. I have
Okay.
survey,
No,
sir.
that's nothing wrong. I'm just asking. Okay. Just go to the other next slide.
Assessment.
So 186, you told that glucose level. Okay. So
Yes.
is it really necessary to give insulin for this patient?
Yes, sir. It is necessary as they cannot bear. No, sir.
They can?
Bear the pain. No, sir. As
Hey, I am asking about blood glucose level. Blood glucose level 186 only. Random, you have been showing that.
yes, sir.
For why can't this drug itself, glibenclamide and metformin itself enough for this patient? Okay, so for this insulin, it's not necessary that to 10 units.
Okay, sir.
So your intervention should be like that. Okay. So
Okay,
you
sir.
may be not aware of these things.
Yes, sir.
Okay, so but anyhow, as a pharmacist, you are coming under pharmacist category. Okay.
Yes, sir.
So we should not forget our profession.
Yes, sir.
Okay. And
Yes.
you all qualified B pharmacy. So you're basically a pharmacist. So
Yes.
that doesn't matter what specialization you have been choosing on your PG. Okay.
Yes. Yes,
Okay.
sir.
Okay.
Thank you, sir.
It's a good case. So pleural effusion is more whatever you given. Very good picture about pleural effusion. That's good.
Thank you.
Yes, thank you.
Thank you, sir.
Thank you, sir. Any more questions from the respected judges? So can we move to the next presentation?
Yes, ma'am.
Thank you, sir. I'd like to invite Vishal Srivastava, sir, from LNCTU University. He posted a presentation on physiochemical extraction and preliminary phytochemical screening of roots of Zooxantheria abhasica leaf case. Vishal,
Yes, ma'am.
sir, am I audible?
Yes,
to
ma'am.
you?
Yeah. Yes, ma'am.
Share your screen. Sir, please share your screen.
Yes, ma'am. Yes, ma'am. I'm trying of this.
Yeah, okay.
Mm-hmm. Is it visible?
No, sir, it is not.
Why it is not a document?
It is visible now, sir.
No. It's not. This is
What?
not. This is not one.
So just take your time. We'll move to the next one, and afterwards we'll call you again.
Just a minute, man. I have shared it and I tried.
Open the slide, then you share.
Sir, can we move to the next one with your permission?
Yes.
Yeah.
Yes.
Thank you. I'd like to invite Parul Ria ma'am from Sheet College of Pharmacy, India. E-poster presentation on effect of both analgesic and anti-inflammatory activity of Butuca frondosa lin leaves on Swiss albino rat. Parul ma'am, are you present there?
Yes, good afternoon all.
Good afternoon, ma'am. Please share your screen, ma'am. No.
I'm not able to see.
हाँ ऐसे बोलोगे चल
Sorry, sir, I can't able to share it right now. May I come after some time?
Okay, ma'am. Okay, ma'am.
Sorry for inconvenience, ma'am.
It's okay, ma'am. So can we move to the next one?
Yes.
Yes. Mummy. Sorry. I would like to invite Deepak Raj Oja, sir, from Sardar Bhagwan Singh University, India, for e-poster presentation on ethanopharmacological evaluation of Cornus captiva wall extract, Robix leaf, and steam extract on STZ-induced type 2 diabetic mellitus in rats. Deepak, sir, are you present there?
Yeah, I'm present here.
So please share your screen.
I'm audible to you?
Yes, sir, you're audible. Adu, usko catch.
Yes.
Okay.
Yes.
Very good afternoon to all and all. Respected dignitaries and session and chair, Dr. K. Karthichan, Dr. Priya Madhavan, Dr. Stanley Thompson, Myself, Deepraj Ojha from M.Pharm Pharmacology of SBS University would like to present my work here. First of all, I would like to focus on the major issue like diabetes and how the leaf and stem extract of Cornus capitata showed significant improvement to treat diabetic complication. Diabetes is a set of illness defined by high blood glucose caused by absolute insulin level. Cornus species have high concentration of terpenoids, flavonoids, and polyphenolic acids. Plants of genus Cornus are effective in treating ailments such as diabetes, cancer, inflammation, and cardiovascular disorder. Therefore, in the present study, the anti-diabetic activity of methanolic extract of leaves and ethanolic extract of stems of Cornus was evaluated. The main region of the methanolic extract of leaves was taken due to this anti-diabetic activity, and the region of taking stems due to their anti-inflammatory property, cardiovascular property, and their healing wound property. Coming to the material and method, the guideline of CAPSA of Government of India will be followed, and prior permission was granted for IEC for conducting experimental study. If we talk about the animal, male Vista rats were taken for the study. The reason of this that the male Vista rat develop more insulin resistance, and in compared to female, it is less or no hormonal. It is less or no hormonal change in male. Talking about the induction, 10% of fructose is given in drinking water for a period of 14 days, followed by a single dose of STZ. As all we know that STZ is widely used to produce pancreatic islet beta cell destruction. It is also used in type 1 diabetic mellitus. It is also used for insulin deficiency and to produce hyperglycemia in rats. The treatment protocol given in this process, diabetic animals were divided into 8 groups, each group having 6 animals. The animal received treatment for a period of 21 days. Group 1, that is the normal control group. In this, the animal received distilled water, that is 1 ml per kg orally. Group 2, that is the toxicant control. In this, the animal received fructose, that is 10% weight by volume for 14 days in drinking water, followed by administration of single dose of STZ, that is 40 mg per kg. Group 3 is given with methanolic extract of CCLD. CCLD, that is Cornus capitata leaf, low dose. In this, the diabetic animal receive a dose of 100 mg per kg of methanolic extract of Cornus capitata leaf. I didn't mention here the group 4, group 5, group 6, but I'll tell verbally that group 4 is given 100 mg per kg of ethanolic extract. Group 5 is given as 400 mg per kg of ethanolic extract, and group 6 is given as 100 mg per kg of ethanolic extract. The parameter we here discuss about the biochemical parameters, hormonal, tissue, and histopathology of liver. Coming to the result section, if we talk about the if we talk about the fast blood glucose level, it can be shown that it can clearly indicate that the administration of methanolic extract of CCLHD and ethanolic extract of CCSD, that is 400 mg per kg body weight for 21 days, have resulted in most significant reduction in glucose level. And if we talk about that postprandial blood glucose level, it can clearly indicate that methanolic extract of CCLSD and ethanolic extract of CCSD, that is 400 per kg body weight for 21 days, have resulted into most significant reduction in postprandial blood glucose level of diabetic animal when compared to the toxicant control, as well as 0 days of treatment. Now coming to the conclusion part. The conclusion that can be drawn from the present study is that cornus capitata have protective effect against diabetic cardiovascular complication. The high dose level of leaf and external extract, that is 400 mg per kg body weight, has shown marked reduction in body weight, BMI, fast blood glucose level, and postprandial blood glucose level in diabetic rat, and caused significant improvement in lipid profile, hormonal level, AST, ALT, CKMB, and CRP level. And thus we can conclude that methanolic extract of leaf causes significant improvement in abnormalities of diabetes cardiovascular complication and can also be explored as potential for management of diabetic complication. It can also be said that phytoconstituents need to be isolated from cornus capitata wall for establishing better anti-diabetic activity with active component. It can also be said that the effect of the leaf and extract of cornus capitata need to be established on various metabolic syndrome related, like diabetes, cardiovascular complication, neurological disease, reproductive system disorder, and others. Thank you. Thank you so much for having me on this presentation.
Thank you, sir. Any questions from the respected judges?
Yes. Yes, the Rajoja.
Yes, sir.
That was very good presentation, late presentation. But one thing is the cornus species have a high concentration of triterpenes, flavonoids, and polyphenolic acid, and already treating ailments such as diabetes, cancer, inflammation, and cardiovascular disorder. So, what make you to choose this plant?
Sir, firstly, the cornus capitata species have a rich content of terpenoids, flavonoids, and as we know that these properties have a great anti-inflammatory properties, cardiovascular property. I think so that from my study I can contribute a particular a drug so that I can assure that in future it can be more beneficial to the diabetic patient. I think herbal formulation are little or less toxic than the allopathy formulation. That's why I choose this cornus capitata species for.
How can you say the Ayurvedic preparations are less toxic than allopathic one?
Sir, majority of literature it has been shown that like just because that most of the allopathic preparation have caused majority of ADRs that we can study in PV, but
not like
in
that.
contrast,
You've been wrongly understood that. Okay, so now herbovigilance is also
yeah,
being
herbo,
in
yeah,
practice.
right, yeah,
Okay,
herbo,
so we
right,
cannot tell that, because why I am saying this is very serious, that herbal preparations along with you taking with allopathic preparations is even more dangerous. Okay, that's why if they take herbal alone, if you, so for example, your result saying that in each and every path there will be a different of active constants. So we cannot measure it. You're just saying this triterpenoids, flavonoids, and polyphenolic acids alone. It's not only the three active ingredients is going to present in this cornus species. It, apart from that, we have so many of active constants that we cannot estimate, and we cannot. That is the process. That is a difficult situation we have been facing, because we have more than a lakh of species. Okay, without identification. Okay, we cannot say that. Okay, so even though we need to be very careful whenever the Ayurvedic or Siddha or natural preparations being given to the patient. Okay, and okay, next. Only the leaves. What of the other extract of stems you're comparing with the leaves and stem parts, right?
right,
Methanolic extract of leaves and ethanolic extract of stems, right?
right.
Okay. So the permission was granted from IAEC.
No,
Where
sir.
is the number? Where is the protocol number?
Sorry, sir, but
approval number.
I didn't mention, but I have this.
Okay.
so that you need to be very careful. You need to mention
Sure,
that.
sure. I'll keep in
Okay,
mind.
authenticated by BSc, that is very good. See, there you given the access number also 422.
Sure, sure.
And I will drop to this. And Carnus capitate, whenever you see some place you mention it correctly. Carnus is a genus name, capitate is a species name. Okay, so some parts you have been given both small letters only.
Right.
Okay. Other things. We can promote the product, but we cannot say that it will be very safer. You have to go more further, more studies, and you have to give acute toxicity studies, chronic toxicity studies, everything we need to develop. Then only we can say it will be a good management for diabetes. Okay. Thank you. Thank you.
Thank you, sir.
What are the other dates?
Thank you, sir. Any more questions from the respected judges?
Ma'am, can I start my presentation? My Vishal Srivastava this.
Yes, sir.
Okay, ma'am. Now it is visible.
Yes, sir, it is visible.
Okay. Sorry for inconvenience. I've, due to the net issue, I'm not able to share my slide. Very good afternoon to all of you. I am Vishal Srivastava, PhD scholar in LNCT University, doing my PhD under the guidance of Viren Sharma sir. Today I am presenting my poster on physiochemical extraction and preliminary phytochemical screening of root of Guzotia abyssinica. Introduction about the Guzotia. Guzotia is a herb present evidently in the every part of the India. Mainly Guzotia is grow for the edible oils and the seeds. It is belong to the family of the Asteraceae. Asteraceae. Commonly it is known as the ramtail and blacktail or the black seeds. It is also known as a nigger. It is used for the antimicrobial activity as well as anti-inflammatory activity. It is also used in the hair loss and cosmetic purpose and as well as in the skin diseases. My evaluation of this plant is for the phytochemical screening, giving the idea about the potential of the potential in pharmaceutical industry. Present study is aimed for the exploration the physiochemical as well as the phytochemical property of the Guzotia. Material and method. That material and method, the collection of the plant root. I have collected the plant root in the local area in the month of the July and December in MP, Bhopal, and authentified by the HOD of the Department of Botany. After the authentication of the plant, I have go for the extraction. For the extraction, I have used 4 different solvents. The solvents are the water, that is distilled water, ethanol, chloroform, and petroleum ether. For this extraction, 5 grams of the powder I have taken and used the Sockless separator for the extraction. For the qualitative analysis of the secondary metabolites, the extract have given and the aqueous as shown in the table. The preliminary phytochemical screening are done for the carbohydrate, glycosides, alkaloids, protein, tannins, flavonoids, fixed oil, and steroids, waxes, and mucilage. As we observe in the table, and that the aqueous extract giving the positive response for the carbohydrate as well as for the protein and it is also give the positive response for the tannin, flavonoids, fixed oils, and the steroids. For the petroleum ether extract, it is only giving the positive response for the steroid and the terpenoids. Then for the chloroform extract, it is also giving the positive response of the steroid and terpenoids. Ethanolic extract giving the positive response for the carbohydrate, proteins, as well as the steroid and the terpenoids. As we are observed for the physiochemical evaluation of root of the Gizotia, that as we are observing that the foreign organ matters are in the graph, it is present about the BA1, and it is the mean of the foreign organ matters, then LOD, and that is the moisture content, then total ash value is I have observed, then acid insoluble ash, water soluble ash, and then swelling index, then and then it is, and then here it is, here I have mentioned that the water soluble extract and the ether soluble extract. The mean value of these parameters are shown in the graph. That graph representing the parameters of the Gizotia abyssinica. From this study from this extraction, I have observed that the aqueous extract has showing that the carbohydrate, proteins, tannins, flavonoids, pig soil, and steroids as the good extract, showing the good extract value, and we can use for the many purpose for determining the different activity of by using this extract. My further planning for after this, I'm going for the CNS activity by using the aqueous extract as well as the ethanolic extract. Thank you.
Any questions from the respected judges?
Nothing, ma'am. Only phytochemical.
Sir, any more questions from the respected
No
judges?
questions, ma'am.
So can we move to the next one?
Yes, ma'am.
Parul ma'am, are you ready with the presentation? Parul ma'am?
Yes, ma'am, I'm here.
Move on to the next person if it is getting late.
Okay, yes, sir, yes, sir. I'd like to invite Ashish Mishra, sir, for oral presentation from Lovely Professional University, Pangwara, Punjab, India. Topic of pharmacokinetic study of fluoxetine alone and its combination with allicin against chronic stress in rodents.
Good afternoon, ma'am and sir. Ma'am, is it visible?
So it is not visible, but it is started sharing.
Now it's okay.
Ma'am, sir, is it visible, sir?
Yeah, now it's visible.
Yes.
Thank you, ma'am and sir. I am here to present my PhD's topic, that is a pharmacokinetic study of the fluoxetine alone and its combination.
Just put it your screen on slideshow. It would be better. Yeah.
Yes, ma'am. My topic is the pharmacokinetic study of fluoxetine alone and its combination with allicin and against chronic stress in rodents. Actually, according to the introduction of the topic, basically the depression is a major issue in this era, because it is a major disability disease that basically affect more than the 260 million people globally, as well as according to the report from the Diagnostic and Statistical Manual of the Mental Disorders, they revealed that the symptomatic characteristic of the depression, as by the cognitive impairment, feeling of guilty, fatigue, weight loss or weight gain, decreased concentration, and recurrent suicidal thoughts. It's mean that approximately whatever the marketed drugs are available for the depression disease, it is also cause a serious adverse effects regarding the aggressive behavior, suicidal risk. So our objective is to overcome the serious side effect that was basically caused by the marketed drugs such as the fluoxetine, as well as the many typical as well as not an atypical antidepressant. Basically, the various despite the better knowledge of the major depressive disorder, there's several mechanisms that basically underlying the depression. Involves the involvement of neurotransmitters such as dopamine, serotonin, as well as the proteins level, means neurotropins such as the brain derived neurotropic factor. According to the neurotransmitter level, that is a dopamine, basically is a catechol neurotransmitter, which also called the pleasure hormone. Basically, that is secreted by the dopaminergic neurons in the human brain. And basically, its function is to regulate the various brain functions involving the learning, locomotion, feeling pleasure, and motivation, as well as along with the dopamine, serotonin, also known as happiness hormones, that involved in the cognition, emotional experience, motor function, mood behavior, as well as other sleep, circadian rhythm, and appetite function. Basically, it concludes that these functions helps to regulate the normal functions of the brain, and fluctuation in these neurotransmitters involves the depressive illness. Means they provoke their decreased level provoke the depressive illness. Along with that, the many proteins are also available for the functioning of the normal brain, for the normal functioning of the human brain. Basically, the brain derived neurotropic factor. It is an important regulator of early neuronal development and survival. Basically, it is required for the late LTP in hippocampal corneoaminoous synapses. According it is basically synthesized in the hippocampal part of the brain, as well as also in the midbrain cortex region, thalamus, hypothalamus, and brainstem. And it, as I told previously, that it maintains the basically it regulates the early neuronal development, that it stimulates the neuroplasticity, that is stable, that is further involved in the stability of the neuronal development. Basically, now our main motto is to reduce the side effect that was caused by the fluoxetine that was used as a marketed drug. Means that it is basically a selective serotonin reuptake inhibitor, because this, as I told previously, that according to the mechanism of depression.
Please move on to your experimental part.
Sorry, sir.
Please move on to your experimental part. Methodology.
Okay, sir.
Yeah.
According to the materials and method, according to my study, I have performed the pharmacokinetic study. Basically, the first is the depression was induced by the chronic unpredictable mild stress disorder. Basically, this procedure will be applied in the rats for the continuously 6 days, basically 6 weeks, basically 42 days, and after that, this means CUMS procedure will be applied to the 8 stressors into the rat, like noise, cage tilting, overlight illumination, food deprivation, water deprivation, crowded housing, soiled cage, and food and water deprivation. After the 42 days, the depressive illness will be was induced in the rat, and further, according to the protocol that was performed after the approval of the Institutional Animal Ethical Committee. There's an experimental protocol. There's a 5-group control group. CUMS means chronic unpredictable mild stress group. Fluoxetine standard group. Allicin, that is basically used as a test group, and it is in the flavonoid component. And second is co-administration of the fluoxetine plus allicin. So these are the assessment of the various depression level using the various parameters. Means the various test was also done to assess the depression level after the induction of the depression through the chronic unpredictable mild stress procedure. There is a sucrose preference test, force field test, actophotometer test, open field test, clonidine induced aggressive behaviors test, and hypnophagia test. These tests will be applied to assess the depression level, then that how much the depression was induced after the COMS model. So according to the result and discussion of the my study, there's an outcome of the HPLC detection for the fluoxetine that was used as a standard drug in the plasma sample, and it was reported that after the 6 hours, the peak concentration as well as the concentration peak time was 13.782. That was reduced to 13.691 after the 2 hours, means 6 to 8 hours. And this is a chromatogram of the blood sample with the retention time of 13.782 plus minus 0.5 minute for the fluoxetine after 6 hours, and this is after 8 hours. But after the result from the outcome of the HPLC detection for allicin, the plasma sample, there was the reduce in the means there's a reduced concentration of allicin in plasma sample as when we compared with the fluoxetine that was a standard drug, but the concentration peak time was 9.89. So next slide, as I told previously, that our motto is to evaluate the effectiveness of the synergism effect of fluoxetine and allicin, because allicin as a herbal drug, when we co-administered with the fluoxetine that was used as a standard drug, they may overcome the side effect caused by the fluoxetine. So after the co-administration of the fluoxetine and allicin, basically there was some time increase the concentration of the concentration peak time, that is from 6 to 8 hours, 13.659 in that's after 6 hours, and 13.894 after 8 hours. So according to the conclusion of my study, the alone the fluoxetine drugs give the high concentration in the plasma, but after the co-administration with the allicin, they may basically their concentration was basically increased. That indicate that they may be a possible alternate and effective therapy against a depressive illness in the future. Thank you very much, sir.
Yes.
Thank you, ma'am.
Have you conducted the method development for this both combination of the drugs?
Sorry, sir.
HPLC method development for allicin as well as
no, sir. Actually, I was performed to the reference article, means to the article.
Article. From the article only you are taken.
So
you are not performed. You have been taken an article and you are performed, right?
Yes, sir.
Okay. 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 Next presentation, please.
Yes, sir. Yes, sir. Thank you, sir. Thank you, ma'am. I'd like to invite Mohammad Shoaib sir from Sheet College of Pharmacy, Varanasi. He posted a presentation on pharmacological aspects of quinine indica lin and its medical properties.
Yes, ma'am.
Mohammad sir, are you present there?
Yes, ma'am.
Yeah. Please share your screen.
It's visible, ma'am?
Yes, sir, it is visible. So please proceed.
Yes, ma'am.
Yeah.
Hello, everyone. Hello, sir. My topic is pharmacological accept of Quisqualis indica lean and its medicinal properties. So it's abstracts my topic on Quisqualis indica. It's a plant or herbal plant. Yes, abstract treatment of chronic disease like rheumatoid arthritis, diabetes, strokes, heart disease, etc. When treated with herbal bioactivities considered as promising and more suitable due to its minimum or no side effects and therapeutics effective treatment. These herbal bioactives as herbal medicine obtained from plant source and vegetable source, so called as natural source. From the past 2 decades, herbalists are using pathogenic agent for the treatment of several chronic as well as acute disease. In this, we had explained briefly about Quisqualis indica lean plant as medicinal plant. This plant has been approved as medicinal plant, but is still used rarely as medicine. So this view contrast on medicinal properties of Quisqualis indica lean. So as one can utilize in medicinal purpose, other important application on this plant as for decoration, ornamental purpose. It's evergreen plant and does not depend upon season to grow and available easily. Quisqualis indica contain phytochemicals such as L-proline, quisqualic acid, L-aspartate, rutin, and two forms of cysteine synthase are as isoenzyme A and isoenzyme B. These phytoconstituents responsible for the various pharmacological activities such as anti-inflammatory, antipyretic, antibacterial, anti-immunomodulatory activities, anti-helmintic activities. Its introduction. The products which are obtained from natural sources such as plants, microorganisms, animal or mineral in the basic need of making drugs used for treatment of diseases, which are synthesized nowadays for making of novel drugs. In the ancient time, the herbal medicine is the only source which are used for the treatment of most of the disease, and today also in many places it have been using for healthcare purpose. So we can say that the herbal medicine remedies is an traditional system of medicine which are used in medicinal practices since from antiquities. Drug the past 2 decades, there has been an increasing interest in the industrialized nation to use medicinal plants. Source of details are pharmacopoeias, indigenous knowledge, scientific literature. So its plant profile by its botanical name Quisqualis indica lean. It belong to Cumbrataceae family and its Quisqualis genus. Its class Magnoliopsida. Its phytoconstituents. Every plant contains several phytoconstituents. In its different part of plants are contained different phytochemical. So its elaboration. Its utilization, traditional use of decoction of root, seed, and fruit can be used as anti-helmintic to expel parasite worms or for elevating diarrhea. Fruit decoction can also be used for gargling. The fruit are also used to combat nephritis. Leaf can be used to relieve pain caused by fever. The roots are also used to treat rheumatism. Flowers are used for relieve headache. The seed of this plant are related species Qui fructus and Qui chinesis. Contain the chemical quisqualic acid, which is an agonist for the AMPA receptor, a kind of glutamate receptor. In flatulence, infusion of leaf is used externally to treat boils and ulcer. Its industrial use, medicinal plant are the richest bioresources of drugs for traditional symptom of medicine, modern medicine, nutraceuticals, food supplement, folk medicine, pharmaceutical intermediates, and chemical entities for synthetic drug. The first step in the value addition of medicinal bioresources is the production of herbal drug preparation using a variety of methods from simple traditional technologies to advanced extraction technique. Its pharmacological activities, the Quisqualis indica link showing various pharmacological activities such as anti-inflammatory, antipyretic activities.
Yes, Mr. Shoya.
Yes, sir.
I am not able to see your slide fully.
Oh, sorry.
You scroll down or scroll up.
It's visible.
No, it's only half part only we are not able to see the results and discussion part.
It's okay, sir.
No. Okay, continue.
So this plant contains some medicinal activities, pharmacochemical constituent, which are responsible for various pharmacological activities. The chemical of microbiologists, agent inflammation is defined as local responses to cellular injuries. This is marked by capillary dilatation,
Go for your results and discussion part. Just you go to results and discussion.
Yes, sir. So these all are pharmacological activities and toxicities. India is sets various toxicities also. India is richly involved with a wide variety of plant having a medicinal value. A number of herbs are thought to be likely to cause adverse effect. So in modern, due to lack of understanding of plant drug interaction having lead of adverse effect reaction that are sometimes life-threatening and lethal. Purity, they are not contaminant example in the form of other herbs which should not be there. And its conclusion. Hello, sir.
Yes, proceed. Conclusion. What is it?
India
Concluded.
is
Okay. Okay. So, how do you selected this plant, this species, antipyretic effect?
so it's with the help of our guide in college and review literatures.
Who authenticated this plant?
So from Department of Department of Botanical in BHU, Dr. KY. I forget
Some
professor
botanists
name.
they identified. Okay.
Yes,
BHU. From
yes,
BHU
sir,
you
yes, sir.
got.
BHU
Yeah,
from head of department, botanical department.
Okay. Okay. So what this is the conclusion? Whether it's been giving the antipyretic effect when compared to the what you compare, what drug you compare with your herbal preparation? What is the standard you used?
So we use in for rheumatoid arthritis for its immunomodulator as an immunomodulator activities and inflammatory activities. In a Vista rat to treat rheumatoid arthritis.
So what drug you used? Standard immunomodulator.
Say indomethacin.
Hmm.
Indomethacin, diclofenac sodium.
Hmm. Okay. So mainly rheumatoid arthritis in this one.
Yes, sir.
Okay. So I'm not able to see the results on discussion. That's why I'm asking you. Okay. Okay. So
Okay.
you should be very clear with your presentation. See, so many things we have for introduction part and all. Okay. So while preparing the slide, it should be highlighted only the activity name. So we should not be no need for all stories because all the points will be very important. But while
Yes, sir.
presentation, we have to concise the things and we need to highlight only the things which is really required to present. Okay. So this
Okay,
is
sir.
the slide not looks good. Okay. So what you have been doing there?
Sam Pharma.
Pharma, second year.
Second year, sir.
You are in project. It's your project.
Yes, sir.
It's your project, huh?
Yes, sir. It's my review article, sir.
Oh, review. Okay. So you have to be very sincere and you need to see while doing your research, you only carrying out the research. No one is doing this research work only. So you are very familiar with this research. So you should be bold enough to express the points very easily. You have to define it.
Yes,
Okay,
sir. I
no
know
need.
it's my first time, sir, so I cannot present very clearly.
Yes, that's why I'm this is my request. Okay, so don't take it as some thing.
Yes, sir. Yes,
Okay,
sir.
so because see the slide itself, it should it should give some good approach. This one thinking about your presentation. Okay, so thank you.
Okay, sir. Thank you
Thank you
so
for
much,
your
sir.
presentation.
Thank you, sir.
Thank you, sir. So can we move to the next presenter?
Yes, ma'am.
Thank you, sir. I'd like to invite Monica Garia, ma'am, from Sardar Bhagwan Singh University, India, for e-poster presentation on modulary effect of age inhibitor, triodoxamine on diabetic complications in streptozotine-induced diabetic in Rett.
Streptozotine, that one, right?
Good afternoon, all of you. Good afternoon, all of you. My name is Monica Garia. I'm the student of Sardar Bhagwan Singh University. My topic is modulatory effect of ACE inhibitor on modulatory effect of ACE inhibitor pyridoxamine alone with the alpha tocopherol on diabetes complication in the streptozotine-induced diabetes in the rat. Firstly, moving to the introduction.
Ma'am, please put your slide on the slideshow.
Okay. Diabetes mellitus is a group of metabolic disorder characterized by the hyperglycemia condition with this elevated blood sugar level. It may be either due to the impaired insulin production or because cells do not properly respond to the insulin that is synthesized from the pancreatic beta cell. Pyridoxamine is a cofactor that functions as an antioxidant preventing the development of ACE. The aim of the study is modulatory effect of ACE inhibitor and alpha tocopherol on diabetes complication in the streptozotine-induced diabetes in the rat. Moving to the material and method, the guidelines of the CPCSCA of the government of the India will be followed, and prior permission was granted from the IACEC for conducting the experimental study. Animals, adult male Vistar rat will be used. Reagent will be used streptozotine, pyridoxamine will be purchased. Alpha tocopherol will be used. Induction of diabetes mellitus. Diabetes mellitus was induced in the adult male rat by the IP injection of the single dose of 65 mg per kg in 0.1 molar citrate buffer pH 4.5. The control group received the equivalent amount of citrate buffer. After 72 hours, blood was taken from the animal. Serum was prepared out and fasting serum glucose level will be checked by the glucose oxidase per oxidase method. Animals showing FSB level above 250 mg per dl were considered as a diabetes and including for the study. Experimental design. The animal will be divided into the 6 group, each group containing the 6 animal. One group of the normal non-diabetic animal received the vehicle 0.1 molar citrate buffer pH 4.5, pyridoxamine 10 mg per kg and alpha tocopherol 10 mg per kg alone and in combination will be administration orally to the different group of the animal for the period of 30 days. In group first, normal animal received the CMC 1% at a dose of 1 mg per kg. In group 2, diabetes animal received the citrate buffer pH 4.5. In group 3, diabetes animal receive a alpha tocopherol alone 10 mg per kg. In group 5, diabetes animal receive pyridoxamine 5 mg per kg and alpha tocopherol 5 mg per kg. In group 6, diabetes animal receive pyridoxamine 7.7 mg per kg and alpha tocopherol 7.5 mg per kg. In group 7, diabetes animal receive pyridoxamine 10 mg per kg and alpha tocopherol 10 mg per kg. The parameter was estimate fasting blood sugar level, creatine, urea, and the uric acid, estimation of oxidative stress, and histopathology of kidney was be. Moving to the result, firstly, if I talk about the creatine level in this graph, creatine indicate the level of creatine was significantly increasing in the staphylococcus diabetes rat, including the kidney damage and induction of the nephropathy. Diabetes animal treated with the alpha tocopherol and pyridoxamine showed decreasing in the creatine level. After the 30 days of the treatment, however, the combination of pyridoxamine and alpha tocopherol at higher dose level caused a more significant reduction in the creatine level in the diabetes as compared to the diabetes animal. As it's known that creatine is the mark of the kidney function and increasing the creatine level indicates some form of kidney damage. Resulting of the study indicate that after treatment the creatine level was be restored near to the normal, indicating the protective effect of pyridoxamine and alpha tocopherol on diabetes neuropathy. Moving to the graph of uric acid and urea. Literature from the previous study revealed that diabetes neuropathy and the level of urea and the uric acid is increased significantly. In this result show the increasing the level of urea and uric acid in the diabetes is indicate that the development of diabetes neuropathy. Treatment of diabetes animal with the pyridoxamine and alpha tocopherol alone cause significant reducing in the level of urea and the uric acid as compared to both of the drug. At lower intimate and higher dose level alone also cause the reduction in the level of urea and the uric acid. However, the combination at the higher dose level produce more significant decreasing in the level of urea and the uric acid. Result of this is in this graph, pyridoxamine and alpha tocopherol produce a beneficial effect in the diabetes neuropathy by decreasing the level of urea and the uric acid and was more favorable to the other drugs. Now moving to the table, moving to the result in present study indicate the staphylococcus cause significant induction of the diabetes in all the group after 72-hour treatment of the all the group. Pyridoxamine and alpha tocopherol caused no improvement in FSG level at 7 days, but at 14 days the animal treated with the pyridoxamine and alpha tocopherol showed a significant improvement FSG level. However, at 30 days the animal treated with the combination of pyridoxamine and alpha tocopherol at higher dose levels showed a significant improvement in the FSG level as compared to the diabetes animal. Animal treated with pyridoxamine and alpha tocopherol alone also caused the improvement in FSG level in diabetes rat. Moving to the discussion indicate that staphylococcus causes significant increasing in the FSG blood as FSG in all the group of normal animal. All groups show the FSG level above 250 mg per dl. Excess increasing in the FSG level may cause the diabetes complication like retinopathy, neuropathy, nephropathy in the animal. Animal showing FSG then 250 mg per dl were taken for the study. Induction of the diabetes complication were confined by the estimation of various parameters in the diabetes rat. Results indicate that steperotoxin induced diabetes animals show significant decreasing in the reaction time when comparing to the normal group. After 30 days of the treatment with alpha tocopherol and pyridoxamine, it was found that there was a more significant increasing in the reaction time in the different group as compared to the diabetes control group. Treatment of these animals with the alpha tocopherol and pyridoxamine caused significant increasing in the reaction time as well as increasing in the jumping response. However, the animal receiving the combination of both the drugs show the more significant increasing in the reaction time. In diabetes nephropathy, the sensory neuron gets sensitized to the external stimuli and the pain threshold is decreased. Treatment will be tocopherol and pyridoxamine improve the sensitivity of neuron. Thank you.
Yes, Monica.
Yes, sir.
Really, it's a good study. What about the other complications? You only seen this one, neuropathy.
Yes, sir.
Is there any other not seen nephropathy and all?
Yes,
Only
sir.
you have been gone for neuropathy, right? Diabetic complications.
Because sir, the agent which use alpha tocopherol have a good effect on
Okay.
neuropathy.
Okay.
That's why.
Able to see that.
Any more questions from the respected judges?
It's a good one. We'll move on to next. Other judges want to put some questions. Excuse me. We'll move on to next.
Yes, sir. Yes, sir. Yes, sir. Thank you, sir. I would like to invite Avindra Kumar, sir, from Street College of Pharmacy for e-poster presentation on hepatoprotective effect of ethanolic extract of Arthrocarpus heterophyllus leaves against rifampicin-induced hepatic ducts in albino rats. So are you present there? Arvindra, sir, are you present there?
We'll call upon next participant.
Yes, sir. I'd like to invite Shreya Singh, ma'am, from up to in College India, e-poster presentation on effects of leaves of Masthanthus arbitris lin on hyperglycemic and glucose-loaded rats. Shreya, ma'am, are you present there?
Yes, ma'am. Yes, ma'am. Good afternoon, ma'am.
Good afternoon, ma'am. Ma'am, please share your screen.
Okay, ma'am. Good afternoon to all. I'm Shreya Singh from State College of Pharmacy, and I'm going to present a poster presentation. And my topic name is Effect of Leaves of Nitranthus arbitris lin on hyperglycemic and glucose-loaded rats. Abstract. Nitranthus arbitris lin is commonly known as night jasmine and parijat.
Shreya, ma'am.
Yes, ma'am.
Please, your slide is not clear. Just make it visible. Yeah, okay.
Yes, ma'am. Nitrensis arbusticis Linn is commonly known as night jasmine and parijat. The plant is a member of Oleaceae family. The shrub is naturally found in tropical and subtropical region of the world. Although it is known as ornamental shrubs, it is rich in medicinal values and pharmacological properties. Nitrensis arbusticis Linn is a native to Southern Asia and is found in northern Pakistan and Nepal. Nitrensis arbusticis Linn grows up to 10 meters. The bark of the plant is dark gray or brown in color and is rough and firm. Leaves are opposite, ovate, and acuminate with margin with the entire and citrate. The petioles are long and hairy and about 5 to 7.5 to 10 mm long with axial concavity. Venation is unicast and reticulate. The lamina is ovate with acute or acuminate apex. The leaves of Nitrensis arbusticis contain arabinoside A, arabinoside B, and C and O, lactanthine, aramine, flavonone glycoside, beta-sterol, and vitamin C. The leaves of the plant are used to cure disease like sciatica, rheumatism, and leaves are also used to treat the chronic fever and internal worms. Properties like laxative, diuretic, and diuretics are present in leaves. Juice prepared from leaves are used as antidote for hepatic venoms and for treatment of various digestive problems. Now we're moving towards the introduction. Plants are one of the most important source of medicine. In India, the Ayurvedic system of the medicine has been uses for over 3,000 years. India's system of medicine includes Ayurvedic, Siddha, Unani, and homeopathic. Herbal therapy provides rational means for the treatment of man's internal diseases, which are considered to be obstinate and incurable in other systems of medicine. It aims to both the prevention and cure of disease. The earliest mention of medicinal uses of plant has been found in Rig Veda, which are written between 4000 to 1600 BC. In Atharvaveda, we find the more varied uses of drug. In Ayurvedic, which is considered as the upper Veda, that definite properties of drug and their uses has been given in general details. Charak Samhita is another earliest treatise on Ayurveda, 600 BC, which listed a total 341 plants produced for health management, and Susastra Samhita also deals with the plant related to medicine. After going to the pharmacological study, we have collected firstly the plant and then goes to the extraction process by the maceration process. Then after that, we performed acute toxicity study, and after that, we decided the dose. Then after that, we performed the pharmacological study, and this is the experiment. First, effect of extract on glucose-loaded rat, and second one is effect of extract on adrenaline-induced hyperglycemia. First, the effect of extract on glucose-loaded rats. In oral glucose tolerance test, glucose 2 gram per kg body weight was given orally to the fasted rat. 2 hours of drug administration and blood sample was collected from the tail veins after 30 minutes, 60, 90, and 120 minutes of glucose treatment. We divided the animals into 4 groups. Group first, which contain the normal group and receive the solvent, 5% gamma acacia. In group second, the glucose loaded group receive the glucose plus solvent. And group third is the test group, receives the glucose plus test drug, 200 mg per kg. And group fourth, this is the standard drug, which receive the glucose plus standard drug, glibenclamide, 0.5 mg per kg. And after that, the experiment design 3, the effect of extract on adrenaline-induced hyperglycemia, in which animals are divided into the 4 groups and of each rats per group. Test group are administered extract of the dose of 200 mg per kg body weight by oral route. Control groups are administered normal saline at the dose of 2 ml per kg body weight. Hyperglycemia control groups are administered adrenaline at the dose of 0.8 mg per kg body weight after 1 hour of drug treatment. Standard group were administered glibenclamide orally at the dose of 0.5 mg per kg body weight. Again, we divide the group animals into 4 groups. Group first, which contain the normal control groups, receive the saline water. And group second, hyperglycemic control, receive the adrenaline 0.8 mg per kg body weight. And group third, test drug group, receives the adrenaline 0.8 mg per kg plus extract of Nitranthus arboristris 200 mg per kg body weight. And group fourth is the standard drug group, which receives the adrenaline 0.8 mg per kg plus glibenclamide 0.5 mg per kg, and blood glucose level was estimated. And the blood after this sample is collected from the tail veins after 30 minutes, 60 minutes, 90 minutes, and 120 minutes, and goes to the result. Result shows the test drug shows the anti-diabetic effect as compared to the standard drug. Then moving to the discussion. In the present study, an attempt was made to establish the effect of plant of Nitranthus arboristris on adrenaline-induced hyperglycemia and glucose-loaded rats. The plant is one of the widely available species in Western Odisha. It was found that the leaves of the plant Nitranthus arboristris can be used in the treatment of many diseases. The oral dose of 200 mg per kg was selected for the study. In adrenaline-induced hyperglycemic rat, the extract is the dose of 200 mg per kg also reduces the blood glucose level significantly in comparison to solvent group. After 5 hours of administration, it also inhibits the insulin release from pancreas and increase the blood glucose level. The result of our study shows that the standard glibenclamide in the dose of 0.5 mg per kg and the extract in the dose of 200 mg per kg reduces the blood glucose level of the glucose-loaded rat significantly. The plant contains the dimannitol, beta-serostrol, flavonoid glycoside, astaglandine, nicotofluoride, oleonic acid, nitranthic acid, tannic acid, ascorbic acid, carotene, etc. These compounds have antioxidant, hepatoprotective, and anti-inflammatory and other actions. Now moving towards the conclusion. Based on the result of this study, it may be concluded that Nitranthus arboristris extract shows interesting possibilities as a source of oral hypoglycemic agent. We believe that this plant must be considered as an excellent candidate for future studies on defining the mechanism and its hypoglycemic activity, as well as for the isolation and identification of active hypoglycemic substance. In summary, the present study demonstrated the administration of ethanolic extract of leaves of Nitranthus arboristris significantly improved systematic insulin sensitivity and glucose hemostasis in normal glycemic rats. Our result supports overall in vivo anti-hyperglycemic activity and activity on impaired glucose tolerance of the extract that proves to be of clinical importance in improving the management of type 2 diabetes mellitus. Thank you.
Shreya, that's a very good presentation.
Yes, sir. Thank you, sir.
So now you're telling that it has to be taken to the clinical level. So how strong you have been saying that? So how in what way it has to be taken to the next level?
Sorry, sir, not visible, sir.
The study, you already proven that the plant has been very effective in treating the blood hyperglycemic levels, right? So which group, glucose-loaded rats or adrenaline-induced hyperglycemia, which one is proved to be very effective?
Sir, both. Sir, both.
Both. So out of that, which is very better?
Yes, sir.
Very better results, going better.
So very better at.
So we need to take to the clinical level, like to preclinical studies or any other thing. We need to fix the dose. Okay, so kinetic studies should be there.
Yes, sir.
So in that level,
yes,
what is your suggestion? Next level. Whether the paper is published, this work,
Yes, sir.
publication completed.
Yes,
The paper is published or not? This work is published or not?
sir, published.
Published. Okay. So then what is your next
sir.
study? You have been saying that it has to be taken to the clinical studies. So in what way you are recommending this? So go for patent. Okay, patent publications or anything.
Yes, sir.
You need to go for patent.
Yes,
Okay,
sir.
so once you get the patent right,
Yes, sir.
then
Okay,
you
sir.
approach some
Okay,
companies
sir.
to help further. Okay, it's not so easy
Okay,
to
sir.
carry out any clinical studies. Okay, on our
Okay,
own.
sir.
Okay, good.
Okay, sir.
Thank you.
Thank you, sir.
Next.
Thank you, sir. Moving to the next presenter, I would like to invite Arnam Chakraborty sir from Sardar Bhagwan Singh University, India, for a poster presentation on evaluation of.
The Parul Rai is there.
Yeah. Yes, sir.
Very good.
Yes, sir, I'm here.
Okay, next will
Very
you be
good.
ready, Ma Parul? Okay. Arnab Chakraborty sir, it's over to you.
Yes, sir.
Yes, yes.
Yes,
You
sir.
go for full view. Presentation mode.
Yes, sir. Is it visible, sir?
Yes. Continue.
Good afternoon, everyone present here. I am Arnab Chakraborty from Sardar Bhagwan Singh University, Dehradun, Uttarakhand, and I am going to discuss about the topic evaluation of hydroalcoholic extract of Grevia asiatica leaves in aluminum chloride induced neurotoxicity in rats. So first of all, we have to know what neurotoxicity is. Neurotoxicity occurs when normal activity of nervous system is affected by some endogenous parameter or some exogenous stimuli, resulting in disruption or neuronal cell death or disruption in the signal transmission. Now, we all know about aluminum chloride. Aluminum chloride or aluminum compounds are therapeutically used in the treatments of hyperhidrosis and also used as antacids. But at a certain level of administration, it can precipitate neuronal toxicity through various pathways, like they can produce reactive oxygen species, which target the polyunsaturated fatty acids present in the cell and converts them to some reactive molecules to produce toxicity. And finally, they cause neuronal DNA damage and cell death. BCL BAX mediated pathway is also involved here because aluminium accumulates in different brain region and induces apoptotic factors like BCL BAX proteins, which increase the mitochondrial permeation and release the cytochrome C. Of this, finally caspase 3 is activated and finally cell death occurs. It's a normal process of cell death, the apoptosis. Neuroinflammation is also done by the induction of NF-kappa B proteins. They decrease the cholinergic function as well by increasing the acetylcholinergic acetylcholine esterase levels and increases intracellular calcium level also. Now, coming to the extraction part, we have collected the dried leaves of G. asiatica from Dehradun, and extraction was done by maceration with ethanol and water at a ratio of 70-30. Literature suggests HEGA or hydroalcoholic extract of Grevia asiatica leaves has high amount of flavonoids and polyphenols, which are responsible for its antioxidant activity. Now coming to the induction part, we used at aluminum chloride at a dose of 100 mg per kg body weight per oral for 14 days. Now, animals which were divided in 6 groups. First of all, the control group, which were administered with normal saline per oral, and animals in the positive control group were administered with aluminum chloride 100 mg per kg. 3 dose levels of hydroalcoholic extract of Grevia asiatica leaves were administered in animals under 3 different groups at a dose of 100 mg per kg body weight, 200 mg per kg body weight, and 500 mg per kg body weight. And there are also a standard group in which the animals are treated with donepezil at a dose of 1 mg per kg body weight per oral. After all, various biochemical and oxidative parameters like calcium level, sodium level, acetylcholinesterase level, LDH level, lactate dehydrogenase level, LPO, SOD, superoxide dismutase, glutathione, they are estimated to know the protective effect against the toxic. Now coming to the results shows a decrease in the level of SOD, GSH, and increase in level of LPO and acetylcholinesterase enzyme in positive control groups, where the standard group shows most significant effect. But animals treated with the extract at a dose of 500 mg per kg body weight decreases in the level of LPO and acetylcholinesterase and increase in SOD and GSH level. Calcium, sodium, LDH level were also decreased in animals treated with the extract at a dose of 500 mg per kg body weight per oral as compared to the positive control. Graphical presentation of my results are given above. And as a conclusion, we can say that due to presence of the flavonoids and polyphenols, the hydroalcoholic extract of Grevia asiatica leaves has shown significant effect in enhancing the oxidative parameter. Calcium, sodium levels are decreased, and LDH level were also decreased. So we can further look for the mechanism of action by which it can show a protective effect on the same. Even we can perform brain histopathology, estimation of other neurotransmitters level present in the brain. Thank you.
So very good work. You're saying that the aluminum chloride is used in the treatment of hyperhidrosis. Okay, yes. So if you are going for aluminum chloride, you are giving the alternative for hypochloride use, or you want to prevent the toxicity happening because of aluminum chloride.
Yes, sir. Yes, sir.
You are just giving the treatment for neurotoxicity, right?
Yes, sir. Right.
Okay. Okay.
Thank you, sir.
That's good. Okay.
Thank you, sir.
So can we move to the next presenter?
Yes, yes, yes.
Thank you, sir. Should I invite Parul ma'am for the presentation, sir?
Yes, yes.
Parul Rai ma'am from Street College of Pharmacy. The poster presentation on.
Yes, ma'am. Good afternoon.
Good afternoon, ma'am.
My name is Parul Rai. I am pursuing my M.Pharm from State College of Pharmacy, Varanasi, India. Objective of my study is evolution of analgesic and anti-inflammatory.
Ma'am, your slide is not clear. Please make it visible now.
Isn't?
Yeah. It's on materials and methods. Yeah, you can do on the abstract, and introduction.
Sorry?
Okay.
Yeah, it is visible, ma'am, now. Thank you.
Okay. Okay, objective of my study is evolution of analgesic and anti-inflammatory activity of Butea frondosa leaf on albino rat. Butea frondosa belongs to the family Fabaceae. Flower of the Butea frondosa is red in color, so that is why it is also known as a flame of forest. In Hindi, it's also called palas. Now, moving forward, the method that used for the pharmacological action of Butia frondosa on analgesic is tail flick method. Tail flick method, the mice were closely restrained in a wire mesh cage and the lower half of the tail dipped in a cold water 4 degrees centigrade. The time for the tail withdrawal from the water was taken as the reaction time. Measurement of the threshold was made 30 and 60 minutes before and after administration of diclofenac sodium. That is, I had taken as a standard doses as 65 mg per kg subcutaneously, and extract of petroleum water and ethanol I had taken as a test drug of Butia frondosa, that is doses as 50 mg per kg, 100 mg per kg and 20 mg, 200 mg per kg oral respectively. Dil- diluted water, that is 10 ml per kg, served as a control drug. Now for same analgesic I has taken is performed for tail immersion method, that is mice was divided into group of 6 animal each. The lowest 5 centimeter portion of a tail was immersed in a beaker of water maintained 55 degrees centigrade temperature. The time in second for a tail withdrawing from a water was taken as a reaction time, with the cutoff time of immersion at a 10 second. The reaction time was measured 1 hour before and 1 hour after oral administration of extracts. And I also given the control drug and diclofenac sodium as standard drug. Then for the anti-inflammatory activity, I had performed carginin-induced palm edema rat, that is the ethanol extract of Butia frondosalin leaf, that is given for 100 mg and 200 mg per kg weight, or and the standard drug indomethacin 10 mg per kg was administered only to differentiated group of rat as shown in my mentioned table result. Acute inflammation was induced half an hour after treatment by subplantar injection. 0.1 ml freshly prepared 1% suspension of carrageenan in a right hind palm of rat. The palm volume was measured in initially and then 1, 2, 3, and 4 hours after the carrageenan injection by the using. Okay, and for the standard drug dosing, I had gone through the OECD guideline 423, and from there I got the doses of the standard drug, and and at the conclusion part, my study showed significant analgesic activity of both ethanoic extract at 100 mg and 200 mg doses in mice, whereas ethanoic extract at 200 mg doses had anti-inflammatory effect. Thank you, sir.
Thank you, ma'am. Any
Which
questions?
method is very effective out of this three analgesic effect you evaluated? No, tail flick, tail immersion, and carrageenan induced palm edema.
Yes, sir.
So which is very effective? Your drug which proves to be.
Sir, tail immersion method for analgesic is more effective at the areas dose of 100 mg.
Okay. So
Yes, sir.
anti-inflammatory? No, not much.
Anti-inflammatory is shown at the carrageenan, but it is on high dose, that is 200 mg per kg.
Okay.
It is also effective, but
Hmm.
not that much.
Okay. Okay, ma'am.
Yes, sir.
Okay.
Thank you.
Thank you, ma'am. Thank you, sir. So can we move to the next one?
Yes, yes, ma'am.
Thank you, sir. I would like to invite Dr. Bandita Datta, ma'am, from Amity Institute of Applied Science, University, Kolkata, India. Oral presentation on multi-target directed drug designing for Alzheimer's and diseases.
Hello, am I audible?
Yes, ma'am, you are audible.
And is the presentation visible?
Yes, ma'am. Yes, ma'am.
Yeah.
And just put it on the slideshow. It would be better.
Yeah, I just did that. Yeah.
Yeah. Thank you, ma'am.
Good afternoon, all and present here, the panelists, the organizers, and all the participants. Today, I would like to talk about the type of research that I do, and I would like to talk more on the Alzheimer's disease. Now, Alzheimer's disease, as we know, is a progressive neurological disorder that causes the brain to shrink and the brain cells to die ultimately. And if we see the data in the world, Alzheimer's disease has started, or it starts right from the age of 20 itself, and slowly it progresses. And as we are reaching towards 2050, the number of cases, the active number of active cases have been increasing exponentially. Now, my research work, generally, we work on these four major parts, that is SARS-CoV, atopic dermatitis, antibacterial activities, and Alzheimer's. We generally do the structure-based drug discovery and the ligand-based drug discovery, generally following the pharmacophore modeling and then experimentally validating the data. The computational data, as usual, we have a databases, we identify the target proteins, see the interactions between our targets, the target protein and the lead molecules. Then we try to do more structure activity relations. We try to modify the structures and then see the activities. Once we do that, once that involves the all virtual studies, we do the designing, and then we synthesize those molecules using a green chemistry protocols. Then we see the drug and the receptor interaction through spectroscopic studies. And finally, we go for the in vivo analysis, although the in vivo analysis generally is done by my collaborators. Now, Alzheimer's disease, we know it has a lot of hypotheses, where the tau hypothesis, that is the tau protein which gets accumulated in the brain, is the mostly studied hypothesis. Apart from that, we have cholinergic hypothesis and amyloid beta cascade hypothesis as well. My group, generally, we concentrate on the cholinergic and the amyloid beta cascade hypothesis. Under the cholinergic hypothesis, now, this hypothesis is present only some 20 years ago only, and it has been found out that between the two neuron cells, the neurons are generated, that is a chemical messenger is being released which binds to the receptors. Now, this chemical messenger which is there is your acetylcholine. Now, in a healthy person, we have acetylcholinesterase enzyme, which is responsible for cleaving the acetylcholine into acetic acid and choline. Now, so there is a kind of equilibrium which is being maintained. The amount of acetylcholine being released and the number gets attached to the receptor, sending the signal to the next neuron cell, like that. But in the case of Alzheimer's affected patients, the activity of acetylcholinesterase is much high. And to address that, we have a WHO prescribed drug, that is the rivastigmine. But the problem with the rivastigmine is that if you see the structure, they have a carbamate moiety. And due to the presence of this carbamate moiety, it causes various side effects or the toxic effects. So we had a plan that we would replace this carbamate moiety with the urea moiety. And then we try to develop the structure, design the structure of the main moiety. Yes, definitely. Rivastic mean we had other drugs like galantamine, donepezil. We tried to do the pharmacophore mapping. On that basis, we had designed this molecule. And this molecule we also designed because it was easy for us to synthesize them. So we can take urea, dimidone, and different substituted benzaldehydes under ultrasonic conditions. We were able to synthesize some 10 derivatives of these compounds. And so these were the different compounds. Now we wanted to change the various groups here, like activating, deactivating groups, electron donating, electron withdrawing groups, just to understand that whether this type of molecules can really bind to the acetylcholinesterase. So we took the molecules, we did the pharmacophore mapping to see how well it fits with the Rivastic min. Then we also optimized the structure. We understood the MESP analysis, the electrostatic structure potential analysis. Definitely, if we are trying to develop it as a drug, then we need to check the ADMET properties, which showed us real good results for this. And finally, to understand the interaction of the enzyme with this molecule, we did the molecular docking. Now, if I talk about this active center of the acetylcholinesterase, generally, this is the active center where our drug molecule can come and bind. And here it's a catalytic triad, which is the most responsible one to which is responsible for cleaving the acetylcholine into acetic acid and choline. So glutamine 334, histidine 447, and serine 203. So our aim was to block this enzyme so that more and more acetylcholine gets produced and can bind to the receptor and can send the signal in the Alzheimer affected patients. So therefore, we did the docking of the acetylcholinesterase with the 10 derivatives that we had designed. And we found that QNZ4 and QNZ10 had given a better binding scores, whereas the same thing was also done with the Rivastic min, which is the approved drug, and it was found that the docking score came much less. So then the same data we validated with the electrostatic potential studies, wherein QNZ4 and 10 we saw that the presence of the polar region and the non-polar region, there was a distinct differentiation between both these groups, which helped us to bind to the pocket in a more firm way. So these two derivatives were really better inhibition to the enzyme. So you can see that once this the derivative, the QNZ derivative fits to the enzyme, the active center or the near around residues interaction we had studied, and we found that from the active catalytic triad, one of the residue was binding with the molecules. Apart from that, the other peripheral anionic site, there were many other tyrosine, tryptophan, aspartate. We were also binding to the molecules, thus stabilizing the molecule and the target protein complex. So seeing this, we feel that this might work in a much better way, and it can act as an inhibitor for the acetylcholinesterase. We also went for the other target, that is the amyloid beta cascade hypothesis, where the beta secretase is the most important target protein here. Now, I will, since short of time, I'll not explain the non-amyloidogenic pathway, but I'll explain what happens in the Alzheimer's affected patients. Now, here what happens is that instead of the alpha secretase, it's a beta secretase which cleaves between the methionine and the aspartate, and then the gamma secretase cleaves the APP protein, that is beta amyloid precursor protein, between the alanine and the threonine. Now, this part of the peptide which comes is called as the beta amyloid, which all binds together to form the neuritic plaque. And this is the plaque which starts getting deposited in the brain cells, tingling the neurons, the nerve cells and all, and that's how the sending of the signal actually gets hampered. Now, for this, we actually thought of following the structure-based drug discovery protocol, and the database that we worked on with this was the phytochemicals. So we generally collected a database of around 1,000 data of phytochemicals. So we had this phytochemical database contained some antiviral, anti-cancer, and anti-inflammatory. That is, this phytochemicals already showed this effects. So we had the lignans, we had the flavonoids, isoflavonoids, majorly. We checked the ADME properties first. We optimized the structures. Then we did the molecular docking. And the top 10 phytochemicals, we had studied the structures where neurocogenin and ruscogenin, both of them showed some really good binding scores. And these two molecules we have given to our collaborators for the in vivo analysis. So in conclusion, I can say that when we targeted the cholinergic hypothesis, we had used the pharmacomodeling, we synthesized the compounds, and they are gone for the further studies. And for the amyloid beta cascade hypothesis, again, here we took the phytochemicals which were already present, available, has shown some activities, and then we tried to see their activity against the beta secretase. And these are the research scholars. Majorly, Aranika worked on that is the beta secretase, and Shornock was the one who worked on the quinazole derivatives preparation and all. And these are the collaborators who are helping me with the theoretical studies or the in vivo analysis, the spectroscopic studies. And that's the department which I belong to. So thank you, organizers, for allowing me to present my paper. Thank you so much.
Thank you, ma'am. Any questions from the respected judges?
So I'm not so familiar with this docking studies, but anyhow, by seeing the effort put up by the presenter, very enormous, and the great work finally. This is very relevant to the Society of Pharmacy or also medical field. Thank you. Is there any other question from other judges? They can put it.
Thank you.
No questions, we can pass on to the next topic.
Yes, sir. Thank you, sir. Thank you, ma'am. I would like to invite Chandrasekhar Chauhan, sir, from RK Pharmacy College, Surrey, Sathanian.
Yes, ma'am, I'm present there.
Yes, sir. E-poster presentation on review on pharmacological activity of Acanthus aspera lim.
Okay, ma'am.
Avanindra is not there, right, ma'am?
Yes, sir.
Okay. Okay. This will be the last.
Yes, sir.
Good afternoon, sir and ma'am. I'm Chandrasekhar Chauhan from RK Pharmacy College, Azamgarh, India. Sir, my
presentation view.
Sir, yes, sir.
Do you use on desktop or laptop for doing the presentation? Everything you
No,
want
sir.
to do it in mobile, huh?
No, sir, I am not laptop. So I want to share my screen.
Okay.
Sir, visible?
For make it more clear, please.
Sir, my topic is a review on pharmacological activity of Acaranthus aspera. Actually, sir, this plant is the very widely in any other countries. Yes, sir. Plant have many has been used as a medicine for thousands of the year. People depend on plant for several purposes like for wood, timber, non-timber, forest product, food and medicine, etc. They have always been used as a rich source of biologically active drugs and have the numerous traditional use. Acaranthus aspera, known as Lagzira chirchira of Marga in the local language. Acaranthus aspera is widespread in the world's tropic and subtropic region of Europe, Africa, Asia, Australia, and America. The therapeutic property of the Acaranthus aspera are mainly due to the secondary metabolites present on it. The most significant of this constitute of the plant alkaloid, tannin, protein, phenolic compound, and flavonoids. Sir, flavonoid so antioxidant property, anti-cancer and anti-inflammatory, same like alkaloid and their derivative, analgesic, anti-spasmodic, antibacterial. Then, sir, saponin chemical constants, just saponin A, B, C, D. They shows insecticidal, antimicrobial activity, and emulsifying, foaming, and hemolytic property. And phenolic compounds show antioxidant, immune enhancer activity, and tannins healing of the wound and inflamed mucous membrane. Occurrences of the various medicinal property like anti-asthmatic, laxative, purgative, anti-allergic, and protective, bleeding, colic, pneumonia, dysentery, headache, and leukoderma. So there is a taxonomical classification. Their kingdom is Plantae, subkingdom is Tracheobonita, superdivision is Spermatophyta, division the Magnoliophyta, class Magnoliopsida, subclass are Caryophyllid or order Caryophyllus, family Amaranthus and genus Acaranthus, and species is Aspera. So there are the many phytochemicals, but I include only three phytochemicals: oleanic acid, ursolic acid, and betaine. So, sir, I include this because I choose hepatoprotective effect. So, oleanic acid, ursolic acid, and betaine are the hepatoprotective effect. But, sir, other phytochemical ingredients like acaranthine, anthocyanin, echidostyrene, acaranthus saponin, A, B, C, and D, oleanic acid, they are the many chemical ingredient present in it. Sir, Acaranthus aspera have the many biological activities like bactericidal, antifungal, spermaticidal, antiparasitic, hypoglycemic, and cancer chemopreventive activity, hepatoprotective activity, anti-inflammatory, anti-arthritic activity, nephroprotective, antidepressant, bronchoprotective, anti-allergic, wound healing, anti-diabetic, immunomodulatory, hypolipidemic activity. Sir, on the behalf of the above data, sir, of the medicine guidance of the Acaranthus aspera used as a treatment of many disorders. Acaranthus aspera further attention and identification of the active constituent responsible for the plant natural activity and demonstrate the mechanism of action which show their hepatoprotective activity. So, sir, I choose Acaranthus aspera root for the purpose of hepatoprotective activity because in the root of Acaranthus aspera contain oleanic acid, betaine, ursolic acid. Thank you, sir.
Thank you, sir. Any questions from the respected judges?
No questions. No questions now.
Yes, sir.
Thank you, sir. Thank you, ma'am.
Thank you.
Avanantra is there? Oh.
Thank you so much, sir. I heartily thank to our session chairs, Dr. Karthik Aryan sir, Dr. Priya Madhavan ma'am, Dr. Telmi Thomas sir, for spending their valuable Sunday with us, and giving their valuable time. Thank you so much, sir and ma'am. There is a small announcement for the participants. I have shared the link in the chat box to download the certificates. There are steps to download these certificates and the new members for existing members also. Thank you so much, everyone.
Thank you, sir and ma'am.
Thank you. Thank you so much. Thank you all.
Thank you. Bye-bye.
On behalf of the Association of Pharmaceutical Research, I would like to express my gratitude to all the speakers and the presenters for their presence and contribution for making this conference more informative and interesting session. APR extends our sincere gratitude to our speaker and participants to take out their time from their busy schedule to grace the events. Thank you so much, everyone.
Okay.